not working, is it? Is that on? Hello? Hello? Hello. It is working. Good. Welcome. Welcome to Bragg Exchange 2025. And it's really great to have a room full of materials researchers together here at the University of Leeds. And welcome to those who are joining on the live stream. Make sure you're here in person next year. It is really nice to have so many people coming to celebrate what we're doing in materials research. It's obviously, as we all know, a very interdisciplinary field. And so it's a really nice opportunity, we hope, for people to come and network and speak together. We have representatives from EPSRC here, from Innovate, from our publishers, from companies. Please do take the opportunity to talk with them as well, explain your research, and see what they could do for you. We have across-campus materials research going across all the faculties, and we have 14 schools represented here, which is really good, showing the interdisciplinary nature. We have 25 external organizations coming in person. Welcome to you all. Roughly 280 people registered to come in person, so I think we'll have people coming and going over the day and a half. And we have posters in the refectory, which Andy will tell you about later. And that, again, has all schools in engineering and physical sciences represented. So it's really great to see the breadth of research across the university and outside the university coming together to celebrate materials science and engineering. A couple of thank yous just before we start. Firstly, to the Royce Institute, which is the National Institute for Advanced Materials, of which Leeds is a founding partner. And they fund quite a lot of what we do in terms of putting on events here. So thank you to the Royce Institute. And also to our sponsors who are listed down the bottom of the screens. And you will be able to see and talk to them at the lunch and the breaks where they have stalls and stands there. So thank you very much to our sponsors. We will say more about the team that supported this, but a large number of people have come together to make this event, and we'll say more about that at the end. But for now, what I'm going to do is pass you over to Andy, who's going to tell you about the practical details of which I know nothing. So Andy, over to you.
Thank you, Edmund. Yes, I get the boring bit. So just some general housekeeping. Fire alarm is tomorrow morning, so we're not expecting anything today. If there is a fire alarm, then please do evacuate the building. And the assembly point is on the grassed area outside the main entrance, so that's that direction as you're leaving. There's lots of staff around who will help guide you if that's the case. First aid, there are plenty of first aiders around in amongst people in the room and also from the staff that are supporting the event. So do let us know if there's any concerns, and we can make all the possible arrangements that need to help with that. Toilets. If you've been in the Simpson building before, you know where they are, but for those who haven't, then the nearest toilets are basically underneath you, underneath the lecture theatre. If you head out that direction, they're straight next to you at the dual doors there. And of course, as has been the case for the past several years, we have to split this across 2 venues. So we are in here for all of the talks and speakers sessions, and then the poster sessions and the exhibition are over in the refectory. So we'll move together as a whole group over at lunchtime, and then there's 3 sessions. So there's a lunchtime now, lunchtime tomorrow, and there's a drinks reception in the evening, all 3 in the refectory. There are posters, there's industry exhibition, and there's a facility exhibition as well of all the Bragg Centre facilities, all there. Just to make you all aware, hopefully you're familiar with this by now, but the Bragg Centre, as part of its public engagement and outreach activities, we live stream the Bragg Exchange. We've done this every year, and it's part of reaching the wider community, the materials community, and making your research as accessible as possible. So just to make you aware that it's the speaker that's being filmed, you may get the back of your head caught on the broadcast, but I'm sure that's absolutely fine. Do let us know if you are concerned, and sit far to the wings if you don't want to be caught on camera. Okay, final few bits. Agenda. You hopefully received the digital agenda when we circulated it around previously. It's also going to be presented on the slides either side, so you can keep up to speed with the upcoming speakers. But we also provide it on the back of your on your name badge. So there's a QR code, you can scan it, but on the back of your name badge as well, there's a QR code to scan and you can get the digital agenda. And then in the 3 sessions that we have in posters, there are poster voting slips in your name badge pack. And all you have to do is basically put the name of the number of the poster that you like the best, drop it in the little box at one of the 3 sessions. Okay. Boring bit done. Now we can get to some of the interesting stuff. And to start, we thought we'd celebrate 5 years. We're coming up to the 5th year now of Bragg. And we thought we'd celebrate by doing something a little creative. And again, as part of our outreach and public engagement activities, we want to reach a wider audience with Bragg and get them inspired by materials. So we thought, what better to do than to get creative and bring in some young talent to help us appeal to a younger audience by making a science fiction film. So I hope you'll join me in enjoying the premiere of Mission Bragg. Okay, a little bit of creativity to kick us off. With that, I'm going to invite Dr. Sean Collins up to chair the first session.
Thank you very much, and very much a big welcome to, I think, what is the materials characterization session this morning. And it really is my pleasure to, I think, introduce our first invited speaker, Dr. Andrew McCluskey from the University of Bristol. Andrew leads a group in the School of Chemistry at Bristol on physics-informed modeling. And I think without too much further ado, a lot of this is working together in close collaboration with synchrotron and neutron sources like diamond light source and ISIS neutron and muon source, but I think I'll hand it over to Andrew and we can hear all about his exciting research. Thank you very much.
Cool, thank you. Oh yeah, I am mic'd up, that works. Cool. I'll do that. All right. Yeah, first of all, thank you very much for having me along. So I started in Bristol like a year and a half ago. Before that, I worked at the European Spallation Source, and before that, I worked at Diamond Light Source. And as both times as a kind of data scientist, and I've just kind of transitioned back into academia. And so just to note, this is daunting because this is the first time I've given like an invited talk. So like I'm being like swift about, and I feel dead fancy in swish, but it's also terrifying. What I'm going to tell you about today is I'm going to tell you about some of the work that I've been doing over what it turned out was the past 6 years in collaboration with some colleagues at the University of Bath. And what we were trying to do was trying to think about how diffusion is estimated from molecular dynamic simulations. And now one of the pieces of work that I might highlight at the end is that we're trying to put this into application at ISIS neutron and muon source. And before I get into that, I just wanted to highlight basically how maybe the vision, if we're going to say that, of my research group. So I run the Statistical Chemical Analysis Methods, or SCAMS for short, research group at the University of Bristol. And what we're interested in is basically this pipeline. So if we have some physical observable that we want to understand, we make some measurement of it. And in making measurement of that physical observable, we turn it into data, right? And then what we do when we analyze our data is we then kind of try and undo the measurement, and we try and understand our physical observable that way. But the thing is, this is what in information theory people would refer to as a noisy channel. We lose information as we go, and what we want to do is, in the SCAMS group, is to try and improve both the measurement and the analysis methods to try and make sure that we get the best possible reproduction of our physical observable. So this is the kind of general vision, but what I'm going to talk to you about today is how we've been trying to do this for molecular dynamic simulations, and in particular molecular dynamic simulations of important energy materials. And so what we're interested in from these molecular dynamic simulations is the diffusion, so the way that the atoms or molecules move in our system. And this is important for a variety of reasons. Diffusion is basically how our batteries work. It's also important in terms of catalysis. If we think about zeolites, if you've got a zeolite with some molecule moving through it, the diffusion rate actually really defines how effective that zeolite could be as a catalyst source. And if we can use simulation to understand these systems, we could start to move towards a more rational design approach where we try and design our materials from our first principles understanding of the system. And this, if we can do this rational design or really understand our systems specifically, then we can start to think about how we use smart, new, cool-sounding methods like machine learning and artificial intelligence to help us design them. But the final kind of motivator for this is that it's sometimes fun to think about scientific problems. It's sometimes fun to not worry about the application and just worry about the problem at hand because it can be really interesting and enjoyable. And the thing that we're going to look at, I'm going to talk about today, is actually about random walks. Because when things are diffusing, the kind of model of diffusion is that the atom or molecule is randomly walking through our system. And so basically, back in 2019, I was a visiting lecturer at the University of Bath, and I had a coffee with my colleague after one of our lectures, and he was like that, oh, I would really like to know about uncertainties in diffusion coefficient estimation. And I was like, that's easy. And 6 years later, we managed to publish the paper. So it definitely felt like a random walk as we were going through discovering totally new things that we didn't really understand before we started to think about them. So the reason that my colleague Ben was interested in this is because in the literature, he sees a lot of this. So Ben is a batteries material researcher, and he sees a lot of people plotting what are referred to as Arrhenius plots. So this is a plot of the logarithm of diffusion against inverse temperature. And now that has a linear relationship if your material is Arrhenius in nature, and then you draw a straight line through that, and you can get information about the activation energy associated with the diffusion. But can anyone see what's wrong with this plot? There's no error bars on the plot. Now, we're all scientists. We all appreciate that with no measurement is perfect. We need to have some statistical uncertainty about our data. And so Ben asked me, I would really like to get error bars on my plot. And I was like, well, there's a dead easy way to do that. You could just run lots of simulations, right? Just run 10 simulations and calculate the standard deviation. There's your error bar. But that's tedious. You don't want to run lots of simulations if you can just run one. And so what we want to do, actually, is we want to run a single simulation or make a single measurement of our system, and from that get the best estimate of the value or diffusion coefficient, right? We want to have a very precise estimate of our diffusion coefficient, and we want to accurately understand the variance, okay? So we want to both get the correct value and know what that error bar looks like. But we only want to do it from a single simulation. So the way that we do this a lot of the time or the way that the batteries materials researchers do this is also used now in biological sciences in a huge variety of different applications. What people do is they run a simulation, and then they calculate the mean squared displacement over a given time interval. So in this is now we're just looking at random walk data, okay, because I love something dead simple that I can play with. So in this simulation, after about 64 seconds, the units are kind of arbitrary, my random walk has moved, I don't know, probably about 350, that should be meters squared. Sorry about that. So it's moved some amount. And what people typically do is they look at this data and they go, right, okay, I'll just fit a straight line to that because I'm following standard Einsteinian diffusion. And I'll calculate what the gradient is, and then the diffusion coefficient, which is the thing I'm interested in, which describes how fast my molecule is moving, is 1/6 of that in a three-dimensional system. And if we do that, because I'm now playing with random walks, I can do that thousands of times really quite cheaply, right? I can do it on my laptop without a huge amount of trouble. So I can do that 1,000 times for 1,000 different random walks, and actually I think this is 4,096 random walks. What I end up with is a distribution of diffusion coefficients, because this isn't an infinite simulation. Each simulation is slightly different, which means I get a slightly different value for my diffusion coefficient. And this makes up this green probability distribution. This is my 4,096 samples. And you can see that that has got some width because it's a normal distribution. Luckily, things all end up as a normal distribution when you're worrying about random walks. And that's all good. We can estimate an uncertainty from that from running thousands of simulations. But I already said we want to run one simulation. Well, luckily, there's statistics textbooks that we can read, and those statistics textbooks tells us there's a formula that we can use, and that formula tells you that the error is that tiny white bar at the top, okay? So from just using the kind of straightforward method that everyone's been using for a while, I've done a really bad job of estimating my variance from a single simulation. But the thing is, this approach, we can already see there's a problem with it. I've already said that error bars are important. In my data, that white line has no error bars. So what if I start to think about error, right? If I start to think about uncertainty. So if I am a particle on a random walk, or actually, we can think about this. This morning, I got up and I went for a run along the canal. If I go for a run, I run 3 kilometers. There's a limited number of places I can reach. Okay, I can only get so far in 3 kilometers, so many different places in 3 kilometers. If I run 30 kilometers, I can get to so many more places. And this tells us that actually our data is what's referred to as heteroskedastic, right? Or it suffers from heteroskedasticity, which has taken me a long time to learn to say. And this heteroskedasticity actually needs to be included in the model that we're using to do our analysis. Previously, what we were doing, we were assuming our data was homoskedastic, which is to say that the variance doesn't change basically as you go along the x-axis. But I've just told you it does, right? Okay, so what if we throw that into our model? What if we include our heteroskedasticity? So this is the one that we saw before. This is our homoskedastic assumption. We can see that we don't do a very good job. If we do the same thing again with our 4,000 random walks, but this time we know the uncertainty, because we've run 4,000 random walks, so we know what we have a very good estimate of the error bars on our data. We can use instead a homoscedastic model, and you can see again, one thing that's interesting is our distribution gets narrower, right? We're getting a better estimate of the true diffusion coefficient from any given single simulation. We're closer to the true value. So that's good, we've improved things. But as you can see, my white bar is still nowhere near my green bar. So that white bar is again using the equations out of our kind of standard statistics textbooks. So this means we're still missing information in our data. We're still missing information in the model we're using when we perform our analysis. And this thing that we're missing can be explained with this slide, but I prefer to explain it by dancing, which is really embarrassing, but I think it works. So I am now a random walking particle. I'm on a one-dimensional random walk. I can either go one step positive. Well, I realize if I put my head down, I get very loud. I can either go one step positive, or I can go one step negative, okay? So if I go on a random walk for 5 steps, so 1, 2, 3, 4, 5, right? Okay, I've now reached the position of -1, okay? If I take one more step, I can only end up back at 0 or at -2, okay? If I take another step, I could only be at -3, -2, -1, 0, +1, okay? So basically all of the information of where you're going to be in the future on this random walk is encoded in where you've been in the past, right? And this actually means that our data is correlated, okay? So the data is correlated when we observe it. And you might have noticed on the previous slide, that previous one was heteroscedastic but uncorrelated. What if we include correlation in our model? Once again, the distribution narrows, so we get a better estimate from a single simulation of our true diffusion coefficient, but also using our off-the-shelf statistical textbook formula, we're able to get now from this one simulation the correct estimate of our variance. So we're now able to just what I'm essentially trying to tell you is we're able to run one simulation and estimate the variance accurately from that simulation as though we'd ran 1,000. But the people who have been listening carefully might have noticed that actually, to get the variance and then to get the true correlation structure, I had to run 4,000 simulations. So to actually get this, I'm having to run thousands of simulations anyway. So where... Okay. Oh. My sounds? Hello? Oh, I'm back. Hey. So what can we do? That was quite dramatic, actually. So what can we do? Well, we can actually go back and think about what is the information that we have in our simulation. So if I have a simulation that's run for 100 seconds, let's say, let's keep things simple, talking kind of seconds and stuff like that, and I want to know how far a particle is moved in 50 seconds. Well, I actually have many observations of that. I have the observation from 0 to 50 seconds. I have the observation from 1 second to 51 seconds, from 2 seconds to 52 seconds, et cetera, et cetera, et cetera. And I show this here for 2 steps over a simulation of 6 steps. You'll see that there's 5 observations of 2 steps for a simulation of 6 steps. But you see that there's 2 of those samples are completely described by the other 3. Okay? So we don't actually have unique samples. All of these samples are kind of interdependent. But luckily, there's actually and so that means if you calculated the variance of your mean squared displacement from over 2 steps from those 5 samples, you would significantly underestimate the variance because you've got these overlapping samples. However, we actually realized that if we normalize by the number of independent samples that we've observed, you get the correct variance out. You get the variance you would expect if you run thousands of simulations. So you can see that it's actually quite a cheap cost. So basically now we calculate the variance from the 5 samples, and we divide it by 3 because that's our number of independent samples. And that actually agrees really well with the numerical simulations that we ran. So that's the variance part, right? So now I'm able to do that heteroscedastic and uncorrelated analysis on just a single simulation and actually get very close to the true variance. But we saw that that's not good enough. I need to understand the correlation structure. So is there a way that I can get the correlation structure from just a single simulation? And it turns out there is, but you have to kind of assume that the correlation structure is gonna, the way it appears mirrors that of a random walk. Your system has to be random walking, essentially, for it to be exactly correct. But for most systems, it's actually a very good estimate, as we'll see. And now this is a bit of a cheat, this image, because I've shown you the numerical, which is the true covariance structure over 4,000 simulations. And I've compared it to the modeled, which is ours. And you can see that they are very similar. And I've kind of cheated because I've not included color bars. But that ratio, there's not a big difference between them. Okay? They agree really well. And the way that we did this is actually I spent like 6 days in front of a whiteboard doing maths. And it's my favorite like SI I've got for our paper because there's like just 2 pages of maths that no one really needs to read unless they're a really keen bean. And so using this, what we're actually able to do is then we can run a single simulation, get an estimate of the variance, and then we're all happy. And so this is what we do now. So we have, this was the same simulation that I showed earlier, but now instead of a single straight line, we have a posterior distribution because we take advantage of Markov chain Monte Carlo sampling to give us an estimate of essentially of the variance in our diffusion coefficient. So you can see that this green kind of shading indicates the potential models that could describe my simulated data. And if we just take the marginal posterior distribution for the diffusion coefficient, we end up with this distribution in green, okay? So this is what the posterior distribution for our diffusion coefficient. And from that, we can take a couple of things. So we can take a mean, because this is normally distributed, it should be normally distributed for a system that is diffusing. So we can take a mean, which is that vertical, now I'm very colorblind and I've not learned what these colors are. Red line, vertical red line, is our mean. And the horizontal purple bar, yeah, purple, is the variance, okay? So those colors are important because they come back up. So these are the 2 parameters that we're going to take. And what we can then do, right, so I'm just showing you one data set. Yeah, cool. You think you might get it right once. We can now do that on 4,000 simulations because we've got 4,000 simulations to play with. So we can apply this analysis approach to many to make sure that we're getting something reasonable. One thing I need to highlight now is I've now, so the paper got accepted last year, but it actually got put online on the 30th of December. So it's actually 25 because it actually got published in the first issue of the next year. So there is a typo in this, but I didn't realize that at the time. So important things, red mean, purple variance. So we could do that for 4,000 simulations, and we could build up a distribution of our means. Okay, so this is the one on the left. Is a distribution of our means is that red histogram. The gray line that's being mapped out over it is the true distribution. Okay, if you analyze all 4,000 simulations, this is the distribution of diffusion coefficients you would observe. The red line is built up from all the individual analyses that we performed. And then that red histogram has a orange bar at the top. That orange bar is the width of the red, or the variance of the red histogram. And what we can see then on the right is all of my individual purple bars from the previous plots are now histogrammed. And they're drawn alongside the variance that's shown on the red. Okay, so the vertical orange line is the same as that horizontal orange line. Does that make sense? All my colors haven't been confusing? Okay. And now you can see our approach, which is taking just a single simulation and analyzing each of them, overestimates the variance in the end. But it doesn't do a very bad job of overestimating. It only overestimates a small amount. And I don't know about you, but I would much prefer my analysis to overestimate my variance than to underestimate it and give me false confidence. And that's exactly what the previous analyses were doing. If you assumed that your data was homoscedastic, you massively underestimated your variance. And so we've actually got a way now that you can use this. You can use this method and get an estimate of your variance from your single simulation and start to put that into your pipeline. But I've just been talking about random walks, and we saw all the cool instrumentation and stuff like that. We want to apply this to real systems that people are actually simulating. So we applied it to lithium lanthanum zirconium oxide dynamic simulations, and generally speaking, we found the same trend. We found that we were able to estimate the variance relatively well with some overestimation. This data is a bit noisier because obviously to generate this, we had to run thousands of, well, I think we ran about 500 lithium lanthanum zirconium oxide simulations. Actually, we didn't, Sam, my collaborator, did. And so this works well for real systems. And so we made it into a Python package. So this is a Python available, if you use Python, it's available on pip, so you can pip install Kinesis. And if you do molecular dynamics simulations, we probably support the outputs that you produce. We can give you a diffusion coefficient with an accurate uncertainty estimation. And actually, we managed to get this published in the Journal of Open Source Software. If anyone does software stuff, I would highly recommend looking at that journal. It's really interesting. So if we come back to this, we've now got our diffusion coefficients with error bars. Great. This is what I wanted all along, because now I can start to play with things in my Arrhenius modeling. And one of the things that we realized, right, is we can actually do a nice trick where we can use, again, our Bayesian Markov chain Monte Carlo. By including the error bars in our diffusion coefficients, we can get an error bar in our activation energy. We can understand how meaningful our estimate of our activation energy is from a bunch of simulations, rather than just quoting single values, as you see quite often in the literature. I'm just going to check, yeah. So one thing, this is now my time for a wee tour to discuss something slightly different, which is these Arrhenius plots drive me crazy. Because everyone, well, not everyone, all of the undergraduate chemistry textbooks tell you if you've got diffusion, or if you want to make an Arrhenius plot, you take the logarithm of the y-axis and the inverse of the x-axis, and then you draw a straight line, and then from that gradient, you can get your activation energy, or your rate constant, or whatever it is that you're using. But that's not mathematically correct to do, because if your data are normally distributed, and you take the logarithm of a normal distribution, your data are no longer normally distributed. Okay, it's now log normally distributed. And that means that when you fit a straight line, you're not applying the correct statistical model, because fitting a straight line actually assumes that your data are normally distributed, and I just told you that they aren't. And so this came up because someone was using Kinesis, and they were like, Kinesis giving me the wrong answer, and they sent me an email about it, and I was like, how are you calculating the diffusion coefficient? They were like, oh, logarithm inverse straight line, and I was like, that's incorrect. The way that you do it is you should actually fit it in the kind of linear space. Don't try and linearize your function, fit it with an actual exponential, because that's where the statistical model is true. And this annoyed me. I'll try and keep my language good. But this annoyed me, and so I wrote a Journal of Chemical Education paper trying to posit that maybe now, when doing nonlinear optimization is so straightforward, we can do it in Excel. Students can do it in Python much better than I can. Maybe we shouldn't always be teaching chemists to linearize functions when it is statistically wrong and produces an incorrect or an unoptimal estimation of the diffusion coefficient. So that's my tangent is done. Just please stop linearizing things, and please stop making me review papers with linearized functions. So now that we've got these variances in our diffusion coefficients, we can start to play games using Bayesian modeling. So I am an enthusiast of Bayesian modeling. When I previously worked at the synchrotron and the neutron sources, I was trying to get everyone on the Bayesian modeling train. I think it's fantastic. It's dead cool, because you can start to do some really cool things with it. So here we have some data from simulations. These are simulations of a superionic conductor. And these were published quite a few years ago now, and the original authors shared the data with us. Unfortunately, I didn't put the attribution to them, but you can ask me, I'll tell you whose it is. And what they said was that they're observing non-Arrhenius behavior in their superionic material, okay? And they did a really elegant thing where they showed what the actual mechanism was and showed why it was non-Arrhenius. But one of the things that you see quite often is people will just fit, instead of if they think their data is non-Arrhenius, they will fit what's called a VTF function. The difference between an Arrhenius function and a VTF function is a VTF function has an additional parameter. So hopefully we're familiar with the idea that you can fit an elephant with 5 complex parameters. So by adding the VTF function, what we wanted to know, adding the additional parameters in the VTF function, is are we starting to overfit our data, okay? And one of the nice things is Bayesian modeling actually gives you a tool for that if you have an estimate of your variance. And that's what we've got, cracking. So what we can do is we can model the Arrhenius data and model the VTF data. But we only do it on some of the simulation. So we apply this, basically we pretend as though we only ran 40 picoseconds of simulation. And we do this, and you can see that 40 picoseconds, It's hard to say because that blue line does kind of go near the one at very low temperature. So it's difficult to say, are we dealing with Arrhenius model, or are we dealing with a more complex non-Arrhenius behavior? So what if we run longer simulation? Longer simulation, your error bars will decrease. You'll get a more confidence in the result. So there was also 140 seconds of picoseconds. So this is basically just the simulation was run for longer. So you see all those error bars have squished down a wee bit, as you would expect. You're running more than twice as much simulation. And you can see quite clearly, to me, it looks very much like the VTF function is correct. One of the nice things that Bayesian model selection, which is the specific tool that we're using, gives us is it gives you the ability to apply numerical values to, oh, that looks better, that looks worse. So it allows you to estimate what's called the Bayesian evidence, and then you can plot the Bayesian evidence as a function of, well, in this case, our simulation length. Basically, as the ratio of our Bayesian evidence for the short simulation and the long simulation, as it increases, there's more and more confidence for the VTF function. And so we can see this here at 40 picoseconds. Basically, we're below that green line, which is the line below that line. There's a paper from Cass and Raftery from like 2006 where they refer to if it's below that green line, the more complex model is not worth a bare mention, which I feel like is a very polite way of saying just ignore it. So at 40 picoseconds, all the way up to about 90 picoseconds, we don't actually have enough information to say our material is non-Arrhenius. Using this approach. Hello? Hello? All right, am I back? I'm back. I just need to switch it off and on again. Cool. Yeah, and so, but if we go to longer simulation, then we get the confidence to apply the non-Arrhenius model using our Bayesian evidence estimation. And this is where we actually see a real-world tool, a real-world application for the fact that we can now estimate the evidence in our diffusion coefficient. So one of the things that we're now looking at is, can we apply this approach to estimating diffusion coefficient, estimating the variance in diffusion coefficients to quasi-elastic neutron scattering. So one of the ways that people will analyze quasi-elastic neutron scattering is to use molecular dynamic simulations. But when they do that, they don't have error bars. So what if we can give them error bars as well? And actually, there's a PhD student started back in September working with me who's looking at this specifically. So what I wanted to tell you today was, it turns out random walks can be fun, and definitely research is a random walk. I wanted to highlight some of the Bayesian modeling methods that can be used to give you confidence in the analysis you're doing. And I don't think this is the end of the story. Unfortunately, 6 years has just whetted my appetite to learn about diffusion. And so there's lots of different projects to keep an eye out in the future. So with that, I'm going to thank everyone for listening to me. Science is not an individual thing. I didn't do this on my own, obviously, as hopefully you all know. It's very important to acknowledge Ben and Sam, who were collaborators throughout this. So Ben is the academic that first approached me with the problem. And then Alex and Josh, who have contributed heavily to the development of Kinesi. And finally, thank you all for listening to me for the last 26 minutes. Cheers.
And I think we have a few minutes for questions. So I'm very happy to, I think we'd be great to have a little bit of discussion after the talk. So are there any questions from the audience?
Yeah, thank you very much. Excellent. So that's what we really need. Uncertainty, it's important. My question is, you mentioned many times molecular dynamics, and the problem is that actually there is a lot of uncertainty embedded in the molecular dynamics itself. So how you deal with that? Because it's maybe not that random data you can get, because there could be like under the surface, you know, like either a potential selection or model selection, already some errors which can be not random, maybe some systematic error. How would you deal with this?
So the approach that I've just spoken about, it doesn't deal with this. This is a lower level problem. One of the ways that and maybe this is because I used to be a facility person, is that if you put garbage into my code, you'll get garbage out. And it becomes then, I mean, we're also looking at how we improve. So that pipeline I showed you, I would love to improve, because measurement includes molecular dynamics. So can we improve how we run our simulations and things like that? But this doesn't really address that. Sorry.
Thank you. Really nice talk. one potential kind of development of this could be to think about self-avoiding walks, which is kind of what people look at for kind of polymer structures. Is that something you've thought about, and how might that affect some of the statistics that you're having to think about with this?
I would love to think about that more, to be honest, because one of the things that I'm curious, so self-avoiding random walks, you often end up with non-diffusive behavior, so you'll have like your thing will go like that. Now, one of the nice things is that the Bayesian modeling approach that we take to estimate the diffusion coefficient, we just dropped in a straight line function. So you could drop in something with a power law quite easily. However, it's then the question of if your underlying assumptions are correct of the thing being pretty much random. So I would love to try and run some simulations and get some weird data out and see if we're able to estimate things correctly, basically using this same approach. But yeah, it's still a question that I want to investigate. Cool, cheers.
Thank you for a really interesting talk and a really novel way of presenting it. So you mentioned the facilities, and it's interesting because I think you said that with the MD you get a result and you don't have variance, you don't have errors for that, but there are also lots of examples for experimental data, particularly at facilities where you get one chance to do the measurement, you don't get a chance to repeat it. So I was interested to hear your view. This approach, could it also be applied to experimental data from scattering, for example?
So one of the things that I always worried about, so when I worked at the ESS, I was an instrument data scientist, which meant I was meant to worry about uncertainties. And yes, I worried about uncertainties. So one of the things that I think that keeps me up a lot, so I used to be a reflectometry person. So those that don't know reflectometry, it's not that cool a technique. Sorry, Christy. But one of the things that terrifies me about reflectometry is how the data are almost definitely correlated that we're measuring in reflectometry. And similar with SANS, it's known for SANS that the data are correlated, and that's why people use Shannon channel theory to try and split up their SANS data. So I think that there is applications for the kind of broader use of this. The specific thing of like thinking about random walks, less so, but worrying about correlations and heteroscedasticity in our data is really important, I think. I mean, already in most scattering techniques, we do assume data are heteroscedastic, but we never worry about correlation. And anytime I see diffraction, I'm like, I want to know what the correlation is in that diffraction profile.
Thank you.
Cool, thanks.
I think just in the interest of time, I think we might draw the questions to an end to move ahead with the schedule. I think there will be opportunities to talk over the course of the day and at poster sessions to talk further about these questions. I know it's great to see further interest, but I think just to keep things moving on the schedule, I think we'll draw things to a close.
Thank you very much, everyone.
And it's my pleasure then to hand over to Professor Rick Brydson, who will be telling us a little bit about his work on crystallization. Many of you know Rick already at the University of Leeds from his many years working in the Leeds Electron Microscopy and Spectroscopy Centre, also heavily involved in the EPSOC National Research Facility in Advanced Electron Microscopy SuperSTEM. But I think without too long of an introduction, I'll hand things over to Rick. Thank you so much.
Thank you. Okay. This is a bit of a change of topic, and it's work that arose from a programme grant, Crystallization in the Real World, that was headed up by Fiona Meldrum from chemistry, who's in the audience. And it's principally the work of Martha and Marianne, who were a research fellow and a PhD student, respectively. This just studies crystallization principally from aqueous solutions, and crystallization itself is a really important purification step and a separation step in a lot of chemical product development. And if we can understand crystallization processes and the mechanisms of them, whether they're what's called classical nucleation, where you form a large enough stable crystal which ultimately then can grow, or a non-classical route where the process goes via an intermediate structure. If we can understand that, we might be able to control it so we control what product comes out, what morphology, shape of the crystals that come out. And in cases where it's not wanted, such as forming scale in a chemical process, we can try and prevent crystallization. So I'm going to concentrate on inorganic materials, particularly calcium sulfate. And calcium sulfate, sort of normal pHs, aqueous pHs, there's two typical stable forms, really. There's a dihydrate, gypsum, and a hemihydrate, or half a molecule of water, basanite. And basanite forms these little needle-shaped structures. Gypsum is more needle-like, maybe thin plates, which have, in some of them you can see twinning in the plates as well. And it's a very important industrial material in terms of plaster of Paris, hydrating to gypsum plaster. And it's also, it forms a lot of scale in industrial processes. But the actual mechanism of a crystallization from aqueous solution of gypsum is still under discussion. So how can we study the crystallization process? So first off, if we want to study the early stages, we need a microscopic technique. And the one we use in particular is electron microscopy. So I'm going to show electron microscopy results, but you could in principle use other microscopy techniques. So how could we study these early stages? Well, we can simply take a drop of the solution, mix some reagents, take a drop of the solution after a particular time point, and just simply dry it on a support grid and film and stick it in the electron microscope. We might think, oh, that's not very accurate, so maybe we want to dry very quickly using vacuum filtration. Or we might want to decide, well, we'll use a chemical to stop the reaction at a particular time point. So we might plunge this aqueous solution into ethanol, or put ethanol on the grid. The other option is we actually fast freeze the sample to turn all the water to amorphous ice, which is transparent if it's thin enough. And then we might either examine it within the ice, the amorphous ice, or we might sublime the ice off under vacuum. So let's have a look. And the main problem is if you do, so here's as an aside, this is the cryo plunge freeze mechanism. So we take a drop on the end of a grid, plunge it into liquid ethane, or we could spray it onto that grid and then plunge it into liquid ethane. But there are methods, these are all developed particularly by the protein crystallographers in biology to study protein structures, but we've started applying them to these crystallization processes. Okay, so we've got these 5 potential methods. The trouble is when you compare the methods in the microscope, you get different results from the 5 methods. So if you air dry or dry very quickly by vacuum filtration, you do see some large gypsum particles that have crystallized out of solution, but you also see lots of sheaves and aggregates of basanite, hemihydrate rods. So you might think, well, maybe the crystallization process is going via basanite to form gypsum, perhaps. So it's a progressive hydration from a hemihydrate to this dihydrate, gypsum. Okay, so that's 2 methods that give a similar result. But we've got to remember, as we're drying, we're increasing the concentration of calcium and sulfate ions in solution. There's supersaturation in that solution, and maybe that's causing basanite to precipitate out in addition to gypsum. And if you take some theoretical data from the literature, we started at 50 millimolar calcium sulfate, and that solution is unstable with respect to precipitation of gypsum. However, as the thing dries and we get to higher supersaturations, you can see this thing, this saturation index, becomes positive for basanite. So as it's drying, basanite suddenly becomes unstable in solution, and that precipitates out. So that's one reason why we're seeing basanite as well as gypsum. If we now try this chemical quenching method, we just see lots and lots of basanite. So it appears maybe that ethanol, in this case, this chemical quenching, favors the precipitation of basanite. And this is kind of well known in the literature. If we do that freezing step, and then we just sublime off the ice, we get a lot of other stuff, like sodium chloride that comes out of solution, but you only see large gypsum crystals and no evidence of these little needles of basanite. And ethanol, if we go back to that ethanol step, is known to promote basanite formation. It keeps basanite crystals stable. The other thing we see with this sublimation step is when we look on the grid, we see these blobs of calcium and sulfur-rich particles. They're not spherical blobs, so they've not been solid in solution. They've actually dried on the grid. And this could be evidence for some sort of dense liquid phase that's forming prior to crystallization. Because if it was a solid in the liquid, it would be spherical. It would minimize its surface energy. And if we go to this final method, prep method, where we plunge freeze the sample rapidly and examine and use a cooling holder for the specimen to keep the thing at low temperature so the ice doesn't sublime off. So we're examining whatever's in solution embedded in this amorphous ice, which is semi-transparent. Then all we see is large gypsum crystals, but at quite long times, talking hours here rather than minutes. And there's no obvious evidence of basanite at earlier times. And we're beginning to come to the conclusion that this cryopreparation method and examination in vitreous ice, amorphous ice, is probably one of the most faithful methods to capture what's happening in solution at a particular time point during a crystallization event. However, it's only a specific snapshot. Now, the other thing people do in the field is they try and examine the liquid directly in the microscope. But because the electron microscope's under a vacuum, liquid would normally just evaporate off in the vacuum. So basically, people have developed very thin liquid cells with little thin silicon nitride membranes that are thin enough to be transparent to the electron beam. And you have a sort of top chip and a bottom chip like this, and it goes on the end of your electron microscope sample holder. And then you can, in principle, flow in two reagents, so maybe a calcium reagent and a sulfate reagent, which will mix, and then hopefully you get crystallization under the window, this little window in the middle, this thing here that's transparent enough to get the electron beam through. However, there are some common problems with this. You're mixing before the window, so maybe the mixing occurs outside the viewing window. We might get crystallization before we go in. And if you do some fluid dynamics modeling, you can see here's the window and the chip here. Here's the two little tubes going in. There's a mixing chamber here. You can see it's already mixed before it gets to the window. So we might expect crystallization to occur there. Secondly, we're extremely confined in terms of the volume here. We're talking about nanoliters rather than, milliliters in a test tube or a beaker. So this confinement, in principle, could affect the process. The third one is flow. So if you model this flow, what you can see is a lot of the high velocity is going around the outside of the chip and not through the window. So the red is the higher velocity. And also, the mixing seems to occur more efficiently around the outsides. One way you can slow this effect down is to use very slow flow rates. So you get diffusive mixing. But then the mixing tends to occur preferentially around the edge of the chip. And the final one is the fact that the electrons can ionize whatever's in your sample, particularly water. And if it ionizes water, you get lots of ionization products. Because it's a liquid, these are very mobile, they can move around very fast and create lots of problems. So they can, in principle, increase the pH, for example, or reduce things with these aqueous electrons. And just to give you an idea, the sort of context here is that the sort of conditions we use, even if we use very low fluencies of electrons, such as you would use to study a protein, for example, that's a dose which is similar to your sample sitting in the middle of a nuclear reactor. So it's not really very nice on the little specimen that you're doing, so it can create lots and lots of problems. Martha tried to study this on a number of occasions, and there was no obvious crystallization in the chip, in the window. However, if you take the chip apart, you might be able to see lots of crystals around the outside. So we've got this problem of things not crystallizing in the viewing window, which is right in the middle here. However, liquid cells we've found are quite good for studying solid-solid transformations in a liquid. In this case, we're starting with basanite needles and we're hydrating them into gypsum. And you can sort of see what happens is that the basanite needles, you can see them, you can see something on the chip to begin with. They tend to dissolve and then you precipitate these large gypsum crystals. So this basanite to gypsum transformation, which is what you would do with plaster of Paris, you'd hydrate it into plaster, suggests that the basanite, the plaster of Paris, dissolves first, supersaturates the solution, and then reprecipitates out. So having something in the viewing window to begin with is really beneficial for liquid cell. Coming at it from a liquid and finding out what's coming out of solution is much more difficult. And the nice thing about these liquid cell chips, you can take them off the end of the holder for the electron microscope and stick them in say a Raman spectrometer. And you have two signatures. So here's the wave number for the vibrations in the Raman spectrum. Basically, basanite and gypsum have slightly different wave numbers for the vibration. And as a function of time, you can follow the transformation of basanite here into gypsum, and here you get a little signal which is due to aqueous sulfate ions, which backs up the idea that we're dissolving the thing first and then it's reprecipitating as gypsum. However, in our liquid cell, we observe the transformation in about 100 seconds. Here, it's 2 or 3 times longer. And that's because the electron beams pushing basanite into solution faster. So there's effects of the beam, basically. If we now do this fast freezing and examine the basanite going to gypsum in amorphous ice, so we essentially plunge freeze it at different times, we actually get a phase transformation from basanite to gypsum, which occurs now in 15 seconds rather than 100 seconds in the liquid cell or 250 seconds in the Raman case. And that, we believe, is because there's no confinement or very little confinement compared to the nanoliter size in your chip. just to finish off, we applied the same sort of thing to crystallization of calcium carbonate. So we did the 5 methods again, and we mixed calcium chloride and sodium carbonate, and then at different time points to see what was happening. So if we do the drying mechanism, what we see is initially these spherical blobs of amorphous calcium carbonate, which at longer times begin to form little crystals of calcite, which is one form of calcium carbonate, and vaterite, which is another structural form of calcium carbonate. If we do this chemical quench to prevent crystallization progressing, what we see, instead of very small amorphous calcium carbonate particles, very large amorphous calcium carbonate particles, in addition, we see a network of very small spherical calcium carbonate particles, in addition to some crystals of calcite in this case. And so the ethanol seems to be pushing this out of solution, these small amorphous carbonate particles. If we do the full fast freezing and examine the thing in amorphous ice, we see amorphous calcium carbonate here, but we also see some evidence for these darker phase regions in the ice, which we believe is due to higher density regions, which could be the remains of a dense liquid phase. So by that I mean a region where the calcium and sulfate ions are concentrating prior to this amorphous calcium carbonate coming out of solution. So this might be the ultimate sort of precursor for the crystallization process. So that's one particular supersaturation level for calcium carbonate. If we drop the supersaturation level, so it's quite small, we actually don't get any amorphous calcium carbonate particles, but we still see these dense regions embedded in the amorphous ice. And we believe that's because we're in this region where it doesn't spontaneously separate. It has to have a fluctuation in concentration to form this phase. So we don't necessarily see this amorphous region, these amorphous particles. So to conclude, I think studying these sorts of dynamic processes, you've got to be very, very careful with sample preparation artifacts. And ideally, you want to correlate different methods. Borrowing techniques from the biologists, such as cryo methods, look quite promising. And these examination of liquid cell samples are still very, very challenging because of all these issues, basically. And just the last slide, basically, that's work being done in the Limass Centre, and after the Bragg Exchange tomorrow afternoon, we're having a relaunch of the facility since we've finally moved into the basement in Bragg, which is going to occur from 3 o'clock tomorrow, and is kindly sponsored by a Tescan company who manufacture a lot of electron microscopes. So if you're free, please come along and join us for this relaunch event. Thank you.
Thank you so much. I think we have time just for a couple of quick questions. If there are any from the audience, I think we could make sure we ask Rick 1 or 2 questions. And I've got one quick one to start, but I do see one up here. While the microphone's going up, you mentioned that you felt like in the calcium sulfate that the full cryo is the most faithful representation. Could you say a little bit more how you determine whether that's the most faithful or not?
I guess trying to correlate with other methods, really, and the fact that you're not confining the system, so it's more similar to what goes on in a natural process, and any damaged products that are created in the ice by the electron beam tend to diffuse less because it's a solid rather than a liquid. So we believe that limitations due to damage of the electron beam are reduced there, although they will still be present.
Annie?
Thank you, Rick, for the presentation. I was just curious about the fact that you found basanite in some of your findings. Could you observe differences between the structures, like basanite polymorphs, or did you perform any XRD measurements with temperature or something to follow this difference, or the dynamics also using this technique?
No, we didn't, and it would be interesting to try that, I think. There's evidence, I guess, when the basanite forms, sometimes it forms these little things that are like wheat sheaves, which suggest one thing forms, and then you get a secondary nucleation of other basanite on that initial basanite crystal. But I guess doing some sort of in situ X-ray diffraction might be kind of useful, yeah, on these systems, on a two-phase system and studying it. That would be a good idea, actually. Maybe we should run a project on that.
Great. Well, I think hopefully people will have a chance to ask more questions and discuss throughout the day and tomorrow. But just to keep things moving as well, I think we're 5 minutes behind, but we'll give you the full time allocated still, so don't worry. I'd like to introduce Dr. Timea Feller from the School of Medicine here at the University of Leeds. And Timea is a Wellcome Trust early career researcher with having done PhD work at Semmelweis University in Hungary, I think. So thank you so much.
Thank you. And thanks for the opportunity to present this work here. I'm really grateful for this. The work is about active and passive microrheology on blood clots. And then the first question, why blood clots and why rheology, what we choose? So normally, physiologically, we have blood clots when we cut ourselves and we need something to seal the wounds to prevent the blood loss. And that's when normally blood clots are made. But there are also pathological conditions when unwanted blood clots are made, and they are blocking the vessels, preventing the supply of blood from the different tissues, so causing problems in the brain and in the heart. So it's an issue if a blood clot is made in an unwanted location and it's too strong. But it's also a problem if the blood clot is too weak, because then parts of it can break off and travel on with the bloodstream, and again closing blockages at other parts of the body, which is also unwanted and life-threatening. So it's quite a complex task. It's not easy to be a good blood clot, and it's even more complicated if we look at the highly variable shear rates of the circulation. So what is mostly fulfilling this task is the scaffold of the blood clot, which is a fibrin network, which is built up from fibrinogen, this molecule which is insoluble in our blood. So why rheology is a tool what we choose is because, very simply put, rheology can describe and assess the deformation and flow behavior of materials, which is exactly what we would need in this situation. So the tool we choose for that is a magnetic tweezer system we here have in University of Leeds, which can do both active and passive microrheology. It's a custom-built system. My task was to make it functional again, and I will show you what it can do right now. So in both of the cases, we have tiny capillaries, and we put it between the magnets. And for active microrheology, we use the electromagnets around the sample so that the blood clot is filled in the capillary. Microbeads are embedded in these blood clots. So with this magnetic field, we can move the beads and test the environment. So it's stress-controlled rheology. The forces are relatively large, but we are limited at high frequencies of measurement because these are electromagnets, so they need time to a couple of microseconds to turn on. On the other side, we also have passive rheology, where we just test the thermal motion, the random motion of these microbeads. We kind of say that it's also stress controlled, but the stress is the thermal motion of these beads as they kick around in the environment. These are relatively weak forces, especially compared with this active rheology. But on the plus side, we are not limited in high frequencies. So the only limit we have is the frame rate of our camera. And in our studies, we could go down to 6,000 hertz, which is pretty impressive. So our aim in this study is that if you look at it, these look to be two complementary methods. So we try to bring these two methods together and just try to complement the results from each of them. So the results we get from active rheology, we move a bead, and it usually has a viscoelastic response because fibrin is a viscoelastic material. So we see a quick elastic deformation and then a viscous creep. And this is the deformation we are measuring, and from that we are calculating the compliance, which is the resistance of the medium against the motion. So we measure this compliance against time. And then there is this paper from Evans et al. With the help of their method, we can calculate G prime and G double prime, so the elastic and the viscous modulus from the compliance itself. And we can do it in a whole frequency range. So the amazing thing I like about this method, that we need to pull the bead only once, but we have all the frequency data for both moduli, as if we would have been doing a frequency sweep experiment, but we don't have to. We just have to pull it once. We usually measure 10 beads in one sample and average them out, and that's how we get the moduli. And I'm happy to present the first results we have in this system since it's back to action. So it is from samples we got from Krakow, Poland, from Dr. Aleksander Synarski. These are patients who had myocardial infarction, and blood was taken 12 hours after this infarction. We had two groups of samples, patients. One patient group didn't have diabetes, and the other group with black had diabetes. And if you just look at these curves, you see that in average, the diabetic group looks to be stiffer in the clots. There is quite a bit of variability, but it's expected because we are talking about patients. So every patient is different, so this variability is expected. But if we look at given frequencies, we can see this difference in clot stiffness quite straightforwardly. And what is interesting, that the clot structure didn't really look different in this sample. So independent of whether the patients have diabetes or not, they are both have dense clot structure. So it's not really about the network structure itself, but rather the materials, mechanical properties, which comes into play here. And the other method, passive rheology, so we just test the wiggling motion of these beads. When we were tracking the random walk of such a particle, we were really happy to see that with using 10-point averaging, it's 6,000 hertz frame rate, we could go down to a quite good space-wise resolution. So I would say about 10 nanometer is the minimum of motion we can track with this method, which we were really happy to see. And then we did the random walkers, which we have heard from Andrew before. So you see the displacement of the bead against time. And as we expect in water, when they are free to move, they are quick to move, they move a lot. But as we increase the viscosity of the medium with using more and more glycerol, then they are moving more and more slowly. What I would like to point out here, so what I did a linear fitting, so that's where we are right now. There is still space for improvement. But when I do the linear fitting, what I see that the coefficient is decreasing, which I expect because this curve should go more down and down because of the increasing viscosity. But the exponent b is not changing, so they have the same slope, kind of, which is what we expect. We just have to wait more in higher viscous environment for the beads to get to the same place, but they can get to the same place. But when we look at the clot, in the blood clot, actually from the motion, we already see that the bead is getting restricted to a given place, which kind of what we expect because the network is growing around it. And this is what we also see from these random motion graphs, that not only the curves are going more down and down because of the increasing viscosity, but also the exponent is kind of decreasing, so they got more flat and flat, which is showing us that they are getting restricted to some clusters, some space-wise, they are getting restricted. And another thing we also liked and disliked about these experiments, that we look to have a hard limit at 10 to the power of -4 square micrometer, which is exactly translates to 10 nanometer, which is the limit of our resolution. So it actually makes sense, but we realize that we can't go under this level, so it is kind of limiting, especially our coefficient. So altogether, when we bring these two samples, I did the active rheology, averaging 2 beads out. It was working. And then I realized I can do it actually during the formation and the breakdown of the clots as well. The breakdown of the clot is happening by enzymatic degradation. So enzymes are coming and chopping up these clots practically. And we can follow it with the active rheology. First, the formation, you can see the elastic modulus increasing and then plateauing when the network is formed. And then this breakdown is happening in this phase. And then G prime, G double prime is competing with each other as the network is breaking down. But we are limited at really early phases and really late phases because it's not really a proper network. And that's where passive rheology will be a great help, because even though the coefficient is plateauing pretty quickly, so we are reaching that limit really quick, the exponent is actually showing us some information. And it's also showing early stages of the clotting, like up to 10 minutes, which we are not able to follow with the active rheology. And also late stages of the lysis, which we also not really good to follow with the active method. And I would also like to point out that the coefficient, if we magnify this, is also kind of showing some changes. It's just like much less than in the beginning or at later stages. So these two methods look to be quite complementary, which is good news for us. So as a summary, we use microrheology, and it has a huge advantage that we have small sample volume, couple of microliter to fill the capillary. Also, we use passive microrheology, and it has quite promising results so far. We are hoping to separately measure the increasing viscosity during the formation and also the cluster formation by separately measuring the coefficient and the exponent. We've seen that active rheology is powerful once the gel is formed, so in the gel phase, whereas the passive rheology can be a really good complementary method for early phases and late phases of clotting and the lysis itself. We were a bit sad to see, to experience that passive rheology won't be applicable to extend the frequency range. We had the hope that with the high frame rates, we can extend these frequencies to higher ones, which would be really relevant for pathological and physiological conditions as well. Likely this won't be possible because we are limited by this 10 nanometer, which I was quite impressed by, but it's still not good enough for fibrin, so it won't be measurable for formed clots, and we won't be able possibly to extend it for fibrin. But maybe for softer hydrogels, it will be also an option. So I would like to thank to all these people, especially Simon Connell, who has been behind this system all along when these people built the whole system as well. I would also like, in addition to these, I would like to thank Ben Johnson for his help, and also for Jean-Chi Liu for her help during these experiments, and thank you for your attention.
Thank you, and hopefully we can also take a quick question or two at the end of the session. I think there's one there.
Hi, thank you for the presentation. I was curious, what are the relevant timescales for you in terms of frequencies for measurements for those different aspects that you spoke about?
Yes, relevant physiologically or
physiologically,
so the shear rates, so the units are similar as the frequency, and actually they translate to these shear rates as well. So we already have a study out. It was not made by me, it was my previous colleague, who already compared venous thrombotic patient who had pulmonary embolism, and he indeed seen a difference, and this frame rate is actually in our scope, so we're just able to see that. And he was able to show differences there. I would pretty much go as high as possible, because what is really interesting is pathological conditions, and they usually, there the vessels get blocked, and we have high shear rates there. So I would say possibly the higher the better, but yeah. Thank you.
So, should we worry about using passive microrheology in a system which is dynamically gelling? Because you're assuming you've got thermal motion, but you've got additional things going on because the molecules are moving around and releasing energy as they gel. So doesn't that give you extra thermal-like kicks?
Yes, possibly yes. So part of the thing we are doing to get over this is using passivated beads. So they have polyethylene glycol chains, so they make them non-reactive with the environment, so they don't get directly bind. But actually, if I go back, so if we look at these curves, as the gelation goes on, the error is like increasing hugely. And this is one of the things that some beads get trapped quite quickly, and some of them are just still freely moving around. So it's kind of a wide distribution. It's just getting like more variable. But probably it's also a tool, so we can make subgroups, and we can also have extra information based on that. So they can serve as like locational test probes.
So there's also heterogeneity in there.
There is a lot.
Great, thank you so much. I think we'll probably draw the talks part of the session to a close. So let's thank all the speakers one more time. And I think that brings the morning's program mostly to a close. I think there's an announcement before we all break up and go over to lunch in the poster session, which I believe is in the refectory, but I'll hand it over to Chris Hammond now for an announcement.
I think it's true to say that William Henry Bragg is not remembered so much for his research work today. I think he's mostly remembered for his work at the Royal Institution, for the Christmas lectures which he gave at the Royal Institution. The first one, the famous one, The World of... Pick the book up. I'm sure you may the very first Royal Institution Christmas lecture given by Faraday himself in 1849. And William Henry Bragg essentially continued this tradition of giving Christmas lectures. He gave four series altogether, and these books, all produced in books now, are available to browse in the refectory, the far end of the refectory. So do browse through William Henry Bragg's 4 sets of lectures. They're still really well worth reading. You'll really appreciate his very simple powers of exposition, which exceeding all others. So do look at those books, free to look at. Thank you very much.
Okay, thank you. So drawing this session to a close, we're going to all head over to the refectory for lunch, posters, and exhibition. Thank you.
Hi. Good afternoon, everyone. Welcome back into the afternoon session of the Bragg Exchange event. My name is Ardian Marina, and I'll be chairing the functional surfaces session. We have 3 presentations, starting first with, and they all look at different aspects of surfaces and in different performance, functionality of materials. The first one is the invite presentation, which will be given by Professor Tomas Polcar. Tomas shares his time between the University of Southampton and the Czech Technical University, and his focus is on surface engineering. So the title of the presentation today, or the focus of the presentation today, will be on the solid lubricants. So chasing the ultimate solid lubricants, and why is that beneficial for industry. So over to you, Tomas.
Okay, yeah, thank you very much. So hello everyone, I'm really happy to see so many people, especially after the lunch. We talk about Faraday here and Bragg and presentation and experiments. Typically people show experiments and we don't do it anymore. We show AI and some sci-fi movies and this. I will show you some experiment. I prepared that here. Because I do tribology and most of you don't know what is it. We don't know either tribologists, but I'll try to explain. So this is my problem. This is a box, and I want to move that box from here to there. So what I have to do, I have to move this. This always works, this experiment, never failed, okay? Now, if I do that, I want to move it. I still can do it a little bit harder, okay? If I do 2, the force will double, more or less. What about if I put 1 million of these? Can I move it by my hand? Can I make solid lubricant, which will decrease friction so much that I will be able to use my hand and move 1,000 tons? 1,000 tons is the biggest stone ever done by people. One stone, it's in close to Baalbek in Lebanon. They made it, but they didn't move it, perhaps because of friction. So my ambition is clear. I want to move this box when I will start to increase the pressure here between the table and the box. Okay, now how to do that? So I split my talk into 3 parts, more or less. So first I will try to look what is the lowest friction of solids and if it is really worth to look for it. So because sometimes it's great to know that the lowest value or property is this, but maybe there is no need for that. So I will show sliding of large lattice mismatch 2D materials, and I will try to explain why 2D materials are so interesting here. The second problem we have here is, can we transform this 2D nanoscale contact, when you have 2D materials, clearly this is nanoscale contact, into 3D? So make it more realistic for maybe one-day application. And I will show what we try to do and how we test that. And finally, I will try to say, okay, all that is nice, but can we use these 2D materials without need to make 2D materials? You know that, for example, in Manchester, they have all institute to make 2D materials, and they are really fantastic. The problem is they are really expensive, and in tribology, you need a lot of them, and we still don't know how to produce them cheaply and effectively. So maybe there is other way. So, and I will look for all that from mechanical engineering perspective. I'm a mechanical engineer. We use hammer and screwdriver. In Southampton, we don't have that fantastic equipment like you have here, guys. So I will show just hammer and screwdriver. Okay, so let's start. What material would you select for lowest friction? And then, of course, what we try to do first is forget our oil. Oil is dirty, although new American president thinks otherwise, but we don't like oil. So we want to use solid lubricants. So there is no need for any extra lubrication, no oils. So what materials we can select? We can select polymers. Polymers are fantastic. Teflon, for example, is very low friction. But these materials are as well very soft. So if you want to put them on the gear, for example, you would struggle because they wear. So what we want material which has extremely low friction, but it's very hard, very, very strong. So, and 2D materials are really excellent candidates for that. Because if you really look for graphene here or molybdenum disulfide here, so what you can see that we have 2 layers, there is no bonding apart from van der Waals forces, which are very weak, And it's obvious that in that direction, if you see this molybdenum disulfide here, if you will move that sheet left, it will smoothly move. Of course, you can discuss now that indeed there is some problem with orientation, but you can kind of orient it in the way that you will get extremely low friction, at least in theory. Okay, so we say this is the material we would like to test, and now we need to do other things. We need to avoid lattice issue. If you have two same materials like graphene and graphene, for example, and if you put them in commensurate like crystal-like, so of course the atoms will kind of sit like that. So to move them from one minimum energy to another, you need to jump. So one way how to avoid that is to rotate. If you rotate graphene a little bit, then you can really, you see the jump is small and you can move very easily. But the problem is that these, if you have small particles, for example, they tend to go to this kind of commensurate system because it's lowest energy, of course. So we try to avoid that. One way how to avoid it is to not to select two same materials, but two different materials. So we select molybdenum disulfide and graphene. Why? Because they have very big different lattice spacing, so they will never kind of get into this kind of commensurate situation, whatever rotation. So we have our materials, and now we need to look how can we really test that. And that's the first problem, because you have 2D materials, and this is another experiment I bring here, is this. It's outdated because it's from morning program, so I can use it. So what we need to take one 2D material, which is thin about 0.3 nanometer, graphene, another in 0.67 nanometers. We need to place them together and slide one over another. Very, very easy to show in the paper, but very difficult to realize when you have really 2D materials. So I will show you how some other people try to do that. Because if we are able to really do it, we will then get almost no friction and definitely no wear. Why no wear? Because if you have 2D material, if you have any wear, it means destruction of your 2D material. So you have to avoid that at all costs. No wear. Okay, that's small summary how people really try to demonstrate how difficult it is. The one which was published a couple of years ago, it was quite an interesting concept. So what they did, that they put a graphite flake, used AFM tip, atomic form microscope. They slide it in the kind of like brute way to break the tip. So they broke the tip here. You can see, kind of randomly, to be honest. And then they were hoping that if there is a small particle, it will kind of like wrap around the tip. And then you have the tip coated by graphene in this case, and you can slide it against other 2D materials, for example. But if you really look how to do it, that you randomly breaking some tips, you need a very likely lot of MSc students to do that, to hit one correct. And then how you can check really if you have really on top correct 2D materials, I doubt it's actually possible. They use some TEM, but difficult to say what they see there. So very likely this is not the best way to measure anything. Another attempt was here when they use some slider. Again, you have AFM tip, and you put some graphite flake just on the top, kind of glue it. How this can stick if it is aligned, again, very, very difficult to say. Moreover, of course, that this apex of the tip, the exact topography is very, very difficult to get. So it's possible to do it, but very, very difficult to do. And another possibility is to use, for example, silicon oxide nanoball and put them and basically grow graphene on them and then kind of slide them, use them as a AFM tip. But again, not something you would do every day, and especially you cannot do it very easily. And there are many examples, I will not go through of them. So it's very, very difficult. So when we saw that, we say, no, we don't want that because we are simple engineers and we don't have AFM and time and students to do that. So the idea was, okay, let's do it differently. And the idea was very, very simple. So we will take very big flake, we will put small flake on top of that and use AFM just to slide it. You see like the box I just showed you before. They are simple-minded engineers. Just to slide it like that, okay? And we did that. So that was the first experiment we did. And we pushed the flake. So we put small flake. This is just illustration here. This is real experiment. This is illustration. So you have AFM tip, and we just push the flake. So we move, there is a friction between the tip and the substrate. In this case, it was, for example, graphene. So we can see there is some force. And then when we hit the flake, there is this increase. And this is actually frictional force. And then we move the flake, you see? And then we can measure really this kind of difference. And this difference is real friction force of 2D material versus 2D material. An important thing is you see that we have only small contact between the tip and the flake. So all the flake is really like 2D to 2D sliding. Minimal effect of any substrate or other things, which is quite positive. And we can measure the friction force. But the problem is that if you look for really friction, how to calculate it, you need to calculate as a gradient. You need to increase the load, as I put the bottles here. You know, that's what we want. We want to put more bottles and see what happens with friction. But how to do this? Because if we push that, so we use another concept, which was actually dragging the flake. So we push it, we didn't push it, but we just press the tip somewhere in the center and then move it. And because the friction between your silicon or silicon oxide tip and of AFM and the flake is higher than this graphene to molybdenum disulfide, you will move it. Basically, what we did is the second experiment is this one. Instead of pushing, we just push it here and move. And now we can increase the load. So we can see the load effect. So this was the second experiment. Again, very, very simple. So when we did this experiment, you can see you can move the flake like that in both setups. So it works. We can measure the force. And if we measure the force, we can calculate the friction. So that's what we did. And then you have here two combinations, molybdenum disulfide and graphene, and molybdenum disulfide versus hexagonal boron nitride, which is basically the same thing, because concerning lattice spacing, this is very similar to graphene. And if we calculate the friction from these points, what we measured as a function of load, we got coefficient of friction about 10 to minus 6. 1 millionth. What does that mean? It means that if I take 1,000,000 bottles of this, maybe not million, 500,000, and then put them there, and the coefficient would be 10 to minus 6, I would be able to push it by hand. Okay? So that's a really decrease. Typical friction here on the floor is 0.3. If it is 0.1, I would struggle to walk. If it is 0.01, I wouldn't be able to walk at all. There's no way to get stability. You can go to even like 4 orders of magnitude lower. So that's fantastic. This is the lowest ever measured, I think, friction. So we published the paper and look what we can do more. But there was another problem which we spotted imminently, is that it a little bit depends how big the flake is, because the problem is that you have two types of friction here. And people typically ignore that, although it's very important. First friction is between the plate and plate, somewhere in the middle. But you have edges, and these edges are very reactive. This is where you really increase the friction, because the edge will kind of drag. So what we did very, very simple things. We split our friction force into two parts. One was related to shear strength, which is like this area contribution, and then what we call edge pinning strength, the perimeter contribution. So what we are saying here, if you have small flake, the perimeter will be dominant, so it will be difficult to move. If you have very big flake, it would be easy to move, because the perimeter effect would be low. And that's what we really show. And we show that edge pinning effect, a little bit surprising, is independent of flake size, but the shear strength drops with area. As a conclusion of this is that it means that if we really increase the flake, because the shear strength drops with area, then the coefficient of friction may be even lower than 10 to minus 6 we measured. But we don't have that big flakes to move, of course. So what we really say, 10 to minus 6 is mostly upper limit of friction. It's possible to go lower. Right. Of course, that we couple it, we have not only hammer and screwdriver. So we use molecular dynamics simulation sometimes as well. So we use them to really confirm the experiment. So basically, we move this flake, and we just were able to see contribution from this area and from the edges, and it perfectly fit basically to our experiment perfectly. As you can see, this drop here and drop there. So we were very happy because now we can predict it for other materials, not only for those we selected for our experiments. Good, so we were able to measure the lowest friction possible. Of course, there are some problems still. One problem, as I said, is that these flakes, if they are small, especially like this one, it's a couple of microns, they like to be in the minimum energy, even if the lattice spacing is different. So sometimes it's difficult to rotate. So when you try to push it here, you see it will move straight. It will not rotate. There should be momentum, but there is no momentum. It just goes straight because it just follows the crystallographic planes. So there is still, although it's much smaller than for example, graphene to graphene, it's still this effect is visible. So maybe we can even lower friction if we find better materials, better material combination. And when we did that, we tried to estimate this angle of rotation, and indeed we find that when we want to rotate the flake, indeed we can provide that initially is not in this kind of the worst in this energy minimum. If it is energy minimum, sometimes it's difficult to move at all. You tend to destroy it instead of moving that or rotating that. You can translate, you can move, but you cannot rotate. So this is another interesting finding because it's important, of course, if you do some nano device using these materials. Okay, another side effect of this ultra-low friction is that because friction is so low, that if you increase the temperature a little bit by annealing, and this is done a lot in microelectronics, for example, if you have small flake, it tends to rotate. So if you have some small angles, they will immediately rotate back to this minimum. So if you want to use some manipulation at high temperature, you can't do that. The nature stops you to do it and will always tend to rotate the flakes. So this is another important finding directly related to friction. So if you have certain angles of rotation from the minimum energy position, the flake at high temperature will always jump back. Of course, it depends how big the flake is. If it is small, it's easy. If it is large, it's more difficult. Okay, now we have the materials and we say it's perfect, but I'm a mechanical engineer. You know, cars and these things moves in macroscopic world, not in 2D. So what we try to use these 2D flakes and move them, transfer them on 3D parts, something like bearing, ball, and use them in macroscopic tribology, macroscopic sliding. And that's the experiment we try here. As you can see here, so this is graphene, which we kind of wrap around the ball with diameter about 2 millimeter, so it's macroscopic big, and then we slide it in macroscopic tribometer to test tribological properties. So it looks fantastic. Of course, the problem is when you try to wrap 2D material around ball, it's not easy. There's no way to do it. No origami will solve this problem, so you will have always this kind of folding, which I will touch later. But if you select the material somewhere in between and you will slide just here, it's more or less perfect, a little bit stretched graphene. And that's what we did. So we glue graphite. From the graphite, we take layer by layer, and then we glue it on the steel ball. And now we try to measure it. So if it works, I don't know. Yes, you see. So you have flake of graphite or graphene, and then against there is a ball, and we slide it and move it. I think this is first ever macroscopic view of real tribological contact of really 2D material versus 2D material. And we try to measure friction. You remember 10 to minus 6. Now in macroscopic world, you expect similar number. So in the first attempt, we got coefficient of friction 6.2. So we try again, different setup of sensors. We got 22. And then the third one, minus 11. Okay, so clearly our sensors are not correct. And the difficulties here we have is because actually the friction is really very low. So we measure anything but actually the contact. You know, any friction in your mechanical system, you need to kind of separate. We try to do it, but typically it seems so far we're failing that. It's very, very challenging from mechanical point of view to measure that. But we try that. But we have the system and we know how to glue the thing, so it's interesting, but it's really far from industry. And when you apply for abstract project, they say, it's fantastic science, but there is no practical application tomorrow. So didn't get the grant. So another problem we have is that this folding of graphene or other 2D materials, as I said, when you do this, you have these problematic parts. And what we realize that, of course, when we are near or we hit them, that we break them. So we try even to use AFM to see. We use AFM and we just measure the friction. And you see that in some cases, we basically break these wrinkles. You produce these kind of wrinkles on the surface because they need to accommodate this change of topography. So what we did that we of course, we studied that. We try to analyze the force you need, and then we apply molecular dynamics to really simulate the movement of tip and what happened with wrinkles when it breaks or when not. But this is another problem which is really stopping you to apply directly these materials in the way I showed right now. So it's a fantastic field, I think, but it's still necessary to go a little bit deeper and understand how to avoid some of these problems or mitigate some problems. Sometimes we have ideas, very often we still don't. Okay, but we still need the application. So what about another way? Formation of solid lubricant in situ. So we want 2D materials, but we don't want to produce them and we don't want to put them somewhere, transfer them, because it's difficult. But the nature actually helps us in very easy way. This is molybdenum disulfide, but it's amorphous, and it's a bulk material. And when you slide this bulk material, in our case, this is like sputtered coatings, about 1 to 2 micron thick, and if you slide that by your finger, you really align the top. You form the crystal. We heard presentation about formation of crystal. You see, this is a very nice example, how we can form them by fingers. And on the top of the surface, you really produce these perfect 2D materials. You see, this is layer by layer stacked, generating low friction. So actually, these materials, they do it in nature. So we don't need to prepare 2D materials in very expensive ALD or CVD machine. We can just make kind of like preparation coating, which is, for example, amorphous, that slide it and you get that. And that's really great, and that's what I will try to finish my talk with this topic. Because we try it, and you see that if we do simulation, if you have amorphous material and you slide it, this is molecular dynamics, not that many atoms, but it's a reactive molecular dynamics, so it's very expensive computationally. You see that after some time, you start to see, okay, in the middle, you start to see that we build the grains here and there, and then after some time on top, you will see that it gets really organized, more or less. And that's what we see in nature. As I said, if it is molybdenum disulfide, it will do exactly that. Using that, we can even predict, when we have model, we can predict energy and temperature needed for that because it's diffusion-driven process and so on. But if you use just molybdenum disulfide, it's not good lubricant for engineers because it's like graphite. It's great, but very soft. And you need something which is hard because we don't want it gets worn immediately. And it would because it's soft. So what we try to do, we try to dope it. So we take this material like molybdenum disulfide and we dope it. We just add some nitrogen in. Or we take tungsten disulfide, which is almost same thing, and we put some carbon in or chromium in, and then we slide that. And when we slide these coatings, we always get the same thing. Very, very low friction, typically like 0.023. And on top, when we look using electron microscopy, you see that we have this nice layered, this is the surface. This is original one, and when we slide on the surface, we produce this very, very nice ordered layers, which is exactly this low friction structure. So here we go, we can produce 2D materials, maybe not absolutely perfect, but well aligned by sliding. We don't need to produce them elsewhere. So that is a really interesting, of course, industrial potential for that. And then we try to explore that. Our key question now, and that's what we try quite heavily, is how we can use simulation to predict the properties. Because to do many experiments is really cool, but it takes a lot of time, and postdocs are expensive, and PhD students are as well expensive. So we try to replace them first by robots, and the second by simulations. These are done still by postdoc, but one day even the postdoc will be replaced and simulations will be done by AI. But what we do here, that basically our idea is, okay, we want molybdenum disulfide, but we want hard coating, so we will put carbon in. So we will have amorphous molybdenum disulfide of carbon, and then we will slide it. I need to put as much as carbon possible, because more carbon, harder material. These coatings are solid lubricant, but they are harder than the hardest steel. The hardness can be above 10 gigapascal. Okay? So when we have the very hard coating, the question is, what amount of molybdenum and sulfur I need to produce this fantastic layer? Is it 10%, 20%, 70%? We don't know. We can try experimentally, but it would be forever. So we try to employ simulation. So we use molecular dynamics. And this is example here, where you have, so we built our model, so it's a rigid part, which is basically carbon. And then we have some mixture of carbon, molybdenum, and sulfur. This is how we build it. Then, of course, we melt it. Then we quench it to get more or less specific structures. Sometimes you get strange structures, and you need to really think if it is real or not and check it with your temp pictures. But we can really play now with content of 75% of carbon, 50% of carbon, and only 25% of carbon. And because we do kind of like concurrent engineering, here we did experiment. This is purely carbon coating. This is this black one. You see that the friction is typically 0.2. Once you start to put a little bit of molybdenum disulfide, you see that the friction really goes down well below 0.1. And you are here like 0.05, which is pretty good. It's close to oil already. So we did the simulation, and we tried to answer the question, are we building molybdenum disulfide or not? So hopefully the video will work. In the first movie, you see that we have a lot of molybdenum disulfide, and indeed, you see almost imminently, I produced molybdenum disulfide here, and then it slides from there because the friction is lowest here. So it will kind of like separate here. If I have 50% of carbon, it's a bit more carbon, you can see that even that, very quickly, by sliding, you produce these solid lubricants, these 2D materials. So as well, 50% is fine, and we know it from experiment. We were not sure about 75%. The experiment showed that friction is still low, but what about our model? And you can see that the model is small and still can be improved, but even in that model, you can see that here we start to produce basically these 2D materials. They are not perfect by far, but clearly the formation is there. So even for very high content of carbon, so imagine you have 75% of carbon and then randomly distributed molybdenum and sulfur, even that is very likely enough to use this tribochemistry to form these 2D materials. And that's our ambition. We want as high carbon content as possible, but still keep this formation of this solid lubricant. So we look for kind of like optimum here. Just for your information about the model, I will not explain much more details here, but this is, we use reactive, we develop force field because that's the problem here, because we don't have them. So we develop force field for molybdenum, sulfur, and carbon, and we publish that. And then we use that force field, which is reactive. So it's reactive, it reacts. So basically, this is the model which really allows us to see tribochemical reactions. Okay, so this is our one effort we have looking for molybdenum disulfide and carbon. Of course, the question is how to relate this with macroscale experiments, which is a big question, and there is no answer yet. Molecular dynamics is still far from any macroscopic experiment. It will take a lot of time to do that. We try to do it to connect it with finite elements, but it's pretty tricky. Okay, and in the last couple of minutes, I still have, I hope. 2 minutes, that's enough. Yeah, so I will show you just this one slide here. What we did, that we dope it by oxygen, which always people in tribology say, don't do it because oxygen is detrimental, you will produce molybdenum oxide. And we test it, and then when we did the simulation here, again we observe that we form these layers, even with oxide. There's no molybdenum oxide. Again, we produce molybdenum disulfide. And when we did it in compare experiments, which is really what is on the surface. This is very EDS scan over like 1.5 nanometer, and this is MD model. We find that we have the same distribution. And now, in this case, we have triple layer. We can identify it from TEM, do simulation, and now connect it with macroscopic experiment. This is the simulation. This is macroscopic experiments. Not only that we get the value, we get the tendency, but as well the value correct. So maybe we are really somewhere where we can start to simulate the problems and answer the question in tribology. And that's all what I want to say today. So conclusion, so many materials to explore, so many things to do. Thank you very much.
Great, thank you very much, Thomas. Very interesting presentation. I'm sure there will be plenty of questions. So. Can I start maybe until?
Oh, there's one.
Until the
I was afraid that I was either so boring or so convincing, so that's.
Well, at least I think everyone knows now what's a tribology test.
Yeah.
So I think we are okay there. Just a quick question, Thomas, before we have the other question. The effect of environment on those experiments.
Yeah.
What environment you need to get to those levels of friction?
Yes, that's interesting. When we did this 10 to -6, it was done in dry nitrogen. Because when you have water, water molecules tend to kind of like dissociate on the surface. It gets in between these 2D layers, and then you increase the friction. Yeah, we have as well some simulations, some papers about that. That's true. But still, as an engineer, I don't need 10 to -6. There is no equipment needing that. I need 10 to -2. And we can achieve that even in air in humid environment.
Yes, thanks for your presentation. My question is about the case study related to the sphere when it is in touch with this flat surface.
Yeah.
Did you consider the effects of rolling resistance when you are doing this kind of experiment?
No.
I mean, it is very interesting for this kind of geometry to calculate this.
Yeah, no, we didn't. We just did pure sliding. So basically you have your ball is covered by the material, and then we slide it like that. We cannot roll because we are not able to cover full ball. We don't know how to do that. We can cover only part. Yeah.
Any other question? Yes, one more question.
Sorry.
Andrew.
Yeah. I know that you wanted to look at 3D materials, but could you have, for the tribology testing, could you have just coated like a disc and done like a pin-on-disc test? You know how you said it was cracking when you...
Yeah, so actually we did that, and we published paper last year in Carbon. We used the disc, we covered the ball with, I would say, many layers of graphene, and then we slide that. And it worked. The friction was 10 to -4 against silicon, because we use silicon as a disc, basically. And it was macroscopic sliding, I mean, like reciprocal 2 Newton load, yeah. So yeah, it is possible to do it, but again, you must be really lucky not to hit any of these wrinkles, because otherwise you break. So it's good for making papers, but not for industry. So we need to improve the way how we wrap the things. Right. Yeah. But we did that, yeah, and published it.
Okay, I think we'll stop here, but Thomas is around this afternoon and tomorrow, so you can speak to him if you have any other questions. Thank you very much, Thomas.
Yeah, thank you.
We'll send you the bill for destroying university property. Okay, so the next presentation is moving on to soft materials, again, staying at the surfaces, interfaces. The presenters, Dr. Seunghwan Lee is from University of Leeds, Mechanical Engineering. Seunghwan started his research career at Houston University and then moved to ETH Zurich and Technical University in Denmark, and moved to Leeds in 2020. So let's say 2023. And the presentation today will be on biointerface.
Right, thank you very much. My talk is also tribology, but tribology of biosystem. And I think the most well-known example of biosystem where the lubrication is considered important is probably the articular joint. Here are two smart materials, namely cartilage and the synovial fluid. They synergistically contribute to lubricate the joint for a lifelong duration, like 70 to 90 years. Much less known, but is covering even broader range of the lubrication duty in the body is done by a soft matter called mucus. This is a slime or hydrogel. So a majority of its mass is from water, but the solid component, especially comprising the solid network, is called mucin, similar name. Mucin is glycoprotein, so along a protein backbone, center part is heavily glycosylated, whereas two termini remain unglycosylated and hydrophobic. And there are two types of mucins. First, membrane-bound, and second, a secreted mucin. And this secreted mucin is the one making mucus, and it is known to be primarily responsible for lubrication in body too. But in some cases, such as ocular system, the membrane-bound mucin is also participating in the lubrication. So in the couple of decades ago, the molecular mucins have drawn a lot of interest for their lubricating behavior, especially because two types of mucins, porcine gastric mucin and bovine submaxillary mucin, they are commercially available, although some other types of mucins were studied too. So to give you some snapshot on those studies, first of all, mucins work particularly good for hydrophobic surfaces because they're spontaneously absorbing onto them in aqueous environment. And in doing that, these two termini are acting as anchoring group, whereas the glycosylated part recruit water. And the type of mucin affects the lubricity very much. For example, BSM is very, very slippery, whereas PZM is not so much at neutral pH. And environmental factors like pH, ionic strength affects the lubricity as well as impurities. So if you're hearing all this, some of you may already think, okay, the mucin's behavior is somewhere like the amphiphilic copolymers, like pluronics or reverse pluronics, except there are a bunch of the charges. Then we can take a moment here to ask an important question. Why do we study the lubricity of the mucin at all? Is it to understand how it works in the body? For that, the way to graft onto the surface here and here is different. Or is it to utilize mucin to lubricate engineering materials? Because some of them is even cheaper than synthetic copolymers. In some simple cases, yes, hydrophobic surfaces in water, it works. But in a little bit more complicated cases, it's not necessarily working. For example, if you want to lubricate articular joint implants, in order to lower friction, wear, lower the wear particles, so ultimately to prolong the lifetime by injecting the lubricant. Pluronics, yes, it lowers the friction forces, but BSM rather increases friction. And the reason is the fluid here is not just the water, it is a serum, which is a model synovial fluid. So there are a bunch of different biomolecules are present there. And pluronics, because of its non-ionic character, it doesn't interact with them. So this is possible, but the BSM probably does and compromise the absorption and the lubrication behavior. So I'm showing this just to show you that the use of engineering application of the mucin for lubricant is not straightforward, and often it's a lot better with synthetic polymers. But even more serious problem is, as I have already mentioned, it is mucus, not molecular mucin, that lubricates our body in most cases. But the studies on the mucus for their lubricating behavior is extremely rare, except that for preclinical testing of some tissue contacting devices like catheter, endoscope, because we have to make sure that the passage of them along the track should be smooth enough. So they the tissue itself with the mucus on top is employed, which makes sense and relevant. Nevertheless, this system is a lot more complicated and is not likely to offer the understanding on the fundamental lubricating behavior of the mucus. But if you talk about the saliva, and if you agree with saliva may be a kind of mucus too, in a broad sense, then we have a completely different story. And first of all, we have a bunch of studies already on their lubricating behavior. And that means that it is not the difference between the saliva and another mucus is not their scientific significance, but maybe a matter of accessibility and availability. To prepare for saliva sample, all you have to do is just to spit, you know. So some mucus model or mimic would be helpful. So in this regard, we thought that the egg white might be an excellent candidate because it's available and it's cheap, and also it's very clean. And this idea was actually initiated by some Canadian group now some while ago. What they did was basically to extract ovomucin from egg white and regenerate hydrogels at varying concentration. And at a certain point, like 1 to 2%, their rheological properties were almost the same with the porcine gastric mucus. Okay, so we just follow the same procedure all the way, confirming the rheological properties, and moved on to carry out a standard tribology test with a pin-on-disc tribometer. So here we have a pin sliding along a track on a disc. And mucus and measure the friction forces, and this hydrogel was used as a lubricant. And the load was kept at 2 Newton, but we changed or we tested a number of different engineering materials. So contact pressure is very different, and so are the coefficient of friction. Some are quite high, some are quite low. But our focus was not the absolute friction force itself, but how much this gel improved or better compared to water alone. And in this regard, none is impressive, as you may agree. And this is a bit disappointing, but not surprising at all, because mucus was not developed in the body to lubricate engineering materials. But still, one thing drew our attention was these first two pairs, the steel on glass and steel on steel, because it's relatively better. And the one thing in common between these two is that they are pretty well wet by both water and this gel, meaning hydrophilic. So we moved on to another tribometer called the mini traction machine, MTM. This was originally developed to study bearings, and where not only sliding, but the rolling component is very important. And the reason why we chose MTM was not because we have any rolling component in the body, although we have some funny expression, the eye rolling, but you know, the rolling in eye rolling is not really rolling in contact mechanics point of view. And the reason is because the rolling contact always provides much lower tribological stresses compared to sliding contact without the change the external load. And this can be done by having separate motor for ball and the disc. But next technical challenge we had was this volume of the pot, which is 35 milliliters. To some people, maybe this is not a big deal, but you know, that means we need 350 milligram of ovomucin for one measurement, which would be disaster. So instead of the gel, we first filled it up with egg white itself, and then compare with a drop of egg white along the track with thin film. And this second approach may be more reasonable because it's known that the thickness of the mucus along the gastrointestinal tract is known to be about 200 micrometer to 800 micrometer only. never flooded. And to our surprise, these two measurements gave almost the same result, which was very nice. Especially the second approach never gives us a meringue at the end, so it's easier to clean up as well. And also back to the gel now, it gives it's a great stability in the friction signals over 5 hours. It's long enough to acquire a standard coefficient of friction versus speed plot. Again, our focus is how much the gel is better than the water alone. So for this pair, we started with a PDMS because mucin is working best at hydrophobic surfaces. But not excellent, but okay level improvement is observed. However, when we switch it to other types of hydrophobic surfaces, like between plastics or plastic and this elastomer, that wasn't as good. Maybe because it was harder and rougher and all this. But most impressive improvement was observed when we switched the PDMS ball to hard, but the hydrophilic steel ball. And at least one order or more than 2 orders of magnitude reduction was observed compared to hydrophobic surfaces. So the key difference is the hydrophilicity of the ball. So to sum up, it's very interesting to us because mucin is in a way only a building block for mucus. But both mucin and mucus show excellent lubricating behavior, but in different ways. For mucin, it works in water, best at the hydrophobic surfaces due to their amphiphilic characteristic. But mucus, there's no bulk water involved. The interface is air to solid. And it works better, not like mucin, at hydrophilic surfaces. And our proposition is the hydrophilic nature of the gel of the mucus wets the surface better and provides a firm adherence there and make sure the slip happens somewhere inside of the gel, not somewhere else. Then naturally, the following question is epithelium hydrophilic? Maybe, maybe not. But one thing agreed in literature is that the two layers of the mucus on the epithelium. Some different terms are used, but first, firmly adhered mucus layer is inviting the more mucus afterwards. And now to show this first layer is membrane-bound mucin or another secreted mucin. But the key point of this study is that natural and engineering material both show excellent consistency in handling the mucus to bring onto the surface for optimum lubrication, which is making the surface hydrophilic. So for the future perspective, afterwards, our other studies and in literature, there are numerous examples of the hydrogels claiming that there are mucus mimics, mucus-inspired hydrogels, and all of. And given that this study, it's very likely most of them will be firmly adhering onto the hydrophilic surfaces. Then what other properties are needed in order to be selected as a proper mucus for optimum lubrication and further? And we'll continue to be able to answer to that question in the future. And thank you for your attention.
Thank you, Seunghwan. I think we have time for 1 or 2 questions. Anyone? Just a quick question. You mentioned the mechanism being boundary lubrication layer. So how thick are those films and why the surface is affecting the formation of that film from mucin and mucus?
We don't have experimental measurement of the film thickness. So strictly speaking, I cannot answer to that question.
So it's a challenge for the measurement session.
Just calculation is telling us if it's homogeneously applied, it is a few hundred micrometer.
Yeah. Okay. Right. Okay. Well, thank you very much, Seunghwan.
Thank you.
Okay, our next presentation in this session is from Dr. Marietjie Ungerer from School of Chemistry. Marietjie started her research career at Northwest University in South Africa and then moved to University of Cardiff and University of Leeds last year. The presentation today is about nanoparticles and for hydrogen production.
Thank you. Thank you so much for this opportunity. So as you've heard, I just moved from Cardiff University. So this is work that's been going on for a couple of years now. We specifically want to look at hydrogen. How does these reactions occur? Hydrogen for storage, hydrogen for all kinds of applications. So specifically, why hydrogen? If you can have a look at that. This gives you an idea for energy capacity versus then how long it takes you to get this energy back. And just to have an idea, like in this blue section is where batteries sit. And this green section is where hydrogen is. And this is now in terms of different technologies, different ways of having it. And you can see this spans quite an energy gap and as well as a discharge, right? So depending on the technology you have, how long does it take to get the hydrogen back for when you need it? So we specifically look at DFT calculations. We want to include different surfaces, different nanoparticles, different reactions that we want to look at, but specifically today I'm just going to talk about the behavior of hydrogen specifically, and on the material that we want to look at is ruthenium. Typically comes in an HCP packing order, but in our case we want to see what would happen if you now have an FCC packing order and what would happen to activity and reactivity. So as I said, we used DFT, we looked at different surfaces before, including palladium and platinum, and we included as well 3 different methods just to cover all bases. From a theoretical perspective, we first wanted to see what this material would give us in terms of a nanoparticle. So this just gives you the fractions of each of these surfaces that may be present in a theoretical nanoparticle. And this shows us that the 111 fraction would be the highest. But we decided to include all 3 just for completion of these reactions. With this, we also explicitly wanted to look at nanoparticles, and for this we then included various ones. The icosahedral, for instance, has mostly, all of them is 111 surfaces. As with the decahedral one, the cubooctahedral has both the 001 and 111 facets, where the cubic is only 001. So we wanted to include all these different nanoparticles to make sure it's not just an artifact of one type, but different surfaces and different facets that's included with this. Interesting with this data that we have found that we experience an inherent magnetic moment. Now, this material is not magnetic, but now that we have these nanoparticles, even of different sizes, suddenly we have a magnetic moment. And we were not sure here, again, is this an artifact of the modeling that we are doing or an artifact of the nanoparticle in itself? And with this, we see it seemed like these values would go down as the particle would get bigger, and that was not necessarily true. The only thing that happened is that this magnetic moment actually moved to the edges of the nanoparticle, but was still present in all of these nanoparticles. And we observed a similar trend for all of them. We decided to use different software in Gaussian. With this, we can consider these nanoparticles more as a molecule. And again, we observed the same, that there is an inherent magnetic moment with these particles. With this, then we could see that it's because of this occupation of the d orbitals. There's a low spin configuration with electron and a high spin. And it seemed particularly with this FCC ruthenium that the high spin was favored. This is particularly good news for the electronic industry, but for us, what would now happen if we still want to do hydrogen adsorption on the surface, and this is happening? So for this, we then added a hydrogen on each of these surfaces or nanoparticles, and then to look at what would happen. In the smallest cases, with both the icosahedral and the cubic ones, we saw that some restructuring of the nanoparticle occurred, meaning that they are metal stable. But the bigger ones did not show the same extent of this. So it means that for subsequent reactions, we would then stabilize them on a different surface, and then redo some of these calculations. Interesting here, though, again, we observed that the adsorption energy was also then affected by the different calculations that we did by forcing some of these calculations in high spin or low spin. And as well, we still observed a magnetic moment. The other interesting thing that happened in this case is that the hydrogen atom also changed its character to hydronium. So it had a more negative character than what we anticipated. And this would affect then subsequent reactions. We wanted to see then, if we observe this, what would happen if we have the same hydrogen, but on an extended surface then? So we did, again, adsorption in different positions on the different surfaces. And again, the adsorption energies were very, very similar. But we did not observe the magnetic moment. What we did observe, though, is that in some cases, the hydrogen atom sticks really well to the surface. So if these adsorption values are really, really high, it means that evolution across the surface would not happen, which could affect your reactions. But the hydrogen still had a hydronium type of character. After this, we wanted to see then, if you work at experimental conditions with the thermodynamics included, what would happen to a surface like this? So if we can look at the graphs on the left, this is then your pressure. Each of these lines is for temperature in terms of the chemical potential of hydrogen. If we then take each of these lines is a subsequent hydrogen adsorption onto a surface in terms of chemical potential and surface energy, and you combine them all together, we will get then this three-dimensional graph. If we draw a line at the top at the pressure that you want to work at, to the line for the temperature, and down, this is typically what you would see with how your surface would behave in those conditions. And that is equivalent to this circle on the graph. So this gives you a very good idea of when you are actually using this type of surface at whatever conditions you want to work at in the laboratory, how would the surface be? And in this case, it seems like the 001 surface would be nearly fully covered with hydrogen. In one sense, for doing a reaction, this is a really good idea because there's high coverage, so thus a reaction can move forward. But if the hydrogen sticks too well to the surface, it also means that poisoning of a surface can occur. We did this for all three surfaces, and as you can see from these graphs then, the 011 also has a really high coverage, where this 111 surface, the one that we expect to have, would have a 0.5 surface coverage. So we can do these kind of reactions and surface calculations for a lot of different types of materials and reactions, but just wanted to show some hydrogen work today. As you can see from this work, that we can have near experimental conditions and then include different calculations with these. It's always a good idea to not include just one type of software, but if you think there's an artifact with your software, to include some of the other work as well. And for that, I thank you.
Thank you, Marietjie. Any questions?
That's very, very interesting work. Have you sort of tested some of these ideas experimentally or planning to do, or you know, there seems lots of interesting things you've observed in the different structures, and can you try and see with them?
Yes, so the previous work that we did with collaborations with South Africa, we have looked at platinum and palladium specifically because we're looking at bimetals. And so we did reactions with water and SO2 on a surface for hydrogen adsorption, and there it matched experimentally. So this work with ruthenium specifically, there's only one paper currently in Nature Communication where they found this material and said they think there is magnetic moment. But so far we haven't found an experimental partner here yet. So we're really looking for someone, if someone's interested to help us. So yeah.
Any other question?
So is there experimental evidence for this transition to FCC as the particles get smaller or the films get thinner?
So the paper that has been published now is to grow it with seed seed formation. So what we think, if we can do a nanoparticle where the inside then of a nanoparticle is an FCC material, which is much cheaper, then just cover the outside with ruthenium, it should be in an FCC configuration then. So the idea is then to test those in the labs. Yeah.
Okay.
There's no papers yet.
I mean, if you could synthesize them, we
Yeah.
could try and look for
Definitely.
that surface reconstruction or whatever.
It could reconfigure back to HCP, as what we've seen with the cubic ones. We think that would happen because the 001 surface, like in the cubic one, is more unstable. But in the 111, which showed to be the most stable one, if you make an icosahedral type shape, it should be stable enough to test them.
Okay, one more question here.
Yeah, thank you. You just mentioned the stability, and isn't it possible that you may have something which is almost not stable, but keeping somehow like FCC, but when you immerse in hydrogen, the presence of hydrogen can switch the structure?
Possibly. Yeah.
Did you test that, or?
We did not.
Okay.
We only included theoretical with the thermodynamics just to see how the surface would behave, but we haven't actually tested it ourselves yet with MD. That is something we want to do.
Great. I think now will be a good time to draw the session to a close. Well, thank you very much to Marietjie and to all the speakers.
Okay, we now have a short comfort break before we then move into the next session. So please don't run anywhere, but toilets are available if you need.
Okay, we're going to start the last session of today before the drinks reception. It's on soft matter. I should introduce myself and Arwen Tyler. So I'm Daniel Reid, this is Arwen Tyler. The reason for introducing ourselves is fairly recently we've been appointed as the new soft matter representatives on the Bragg Centre committee. So that's good news. And so for those of you who are soft matter people at Leeds, we obviously have some work to do now. And one of the things we'd like to start to do is to arrange some meetings between the community here just to work out how we can help. So watch this space for that. But without further ado, I need to introduce our first speaker. So basically, I'm going to just introduce the first speaker, then Arwen's going to do the rest of the hard work for the rest of the session. So I'm going to introduce the first speaker. Very delighted to introduce to you Wilson Poon from School of Physics at Edinburgh. Wilson has many interests. I guess his background's colloid science, food science, soft matter kind of areas, but he's got wide interests, including I guess history, philosophy of science, that kind of thing, faith and science. So I'm giving an amazing talk quite recently on medieval manuscript. So unfortunately, he can't talk to us about that today. But today, what are you going to talk to us about, Wilson? I'll let you. I'm waiting for that to tell me as well, because I've not got. It's up there. Okay, ductile to brittle transition in jam suspensions, making fudge. Wilson, I'll hand over to you.
Right, is this working? Yeah. So I did something highly unusual this time because this is on Zoom. I wrote my slides 2 weeks ago. Disadvantage of that is that when you discover a mistake, when you're sitting at the back, going over them again, you can't change it. So there's a confusion between 0 and infinity in there somewhere. So there's a power to the -1 missing. But I'll tell you when it comes to it. I hope this only happens once. That's a trick, right? Oh, I do actually have to advance the first one. This is 0, is it? Okay.
This is 0.
Ah.
Again.
Oh, there you go. Right. Okay, so I'm going to talk to you about something called a ductile to brittle transition. And in my case, it happens in suspensions that are jammed. But the material that I'm going to work with is mostly fudge. But I'm going to tell you what it's got to do with the sinking of the Titanic. The first time I gave this talk, people said to me, was the captain eating fudge and not paying attention? That's not the relationship. Okay, so as usual, I'm the guy giving the talk, but these are the people who did the work. In particular, we had a fantastic undergraduate this year doing a final year project with us. So the ductile to brittle transition is what the non-soft matter half of a place like the Bragg Exchange might know a lot about, because that is what happens in metal bashing. So in metal bashing, people know that metals, if you pull a piece of metal, they can fracture either if you plot stress against strain or force against displacement. You can either break the specimen in a brittle way, in which case it goes up elastically in a straight line and it just breaks. Or it can go up and it goes. It's an excellent word that is not in the OED yet. We spell PH, ow. Okay, so it just goes like this in some pathetic way and it just goes plastic deformation. So it fractures totally differently. The surfaces, if you look at them by eye, look totally different. And so another way of showing it is that ductile means if you take a rod and you bend it, it gradually bends, and if you brittle, it just breaks. And of course, this goes, metals are brittle at low temperature and ductile at high temperature. That's why ironmongers always have a big fire in their workshop, because when you're beating out a sword, you don't want the thing to do this, you want the thing to just deform. Okay, and typically, this happens at some higher temperature than room temperature. And in fact, if you do this experiment, the surface doesn't always just show one kind of fracture. Typically, metallurgists look at them and you can see that they in fact are two kinds of fracture surface here. And you can say that the material shows so many % of shear fracture. And what they mean by that in metallurgy textbook is the percentage of ductile fracture, the percentage of the surface that looks like this kind of smooth fracture texture. And what's that the sinking of the Titanic? That's the Titanic being built in Belfast. You can still go and stay in a hotel where they reconstructed one of the lounges of the Titanic. So this is the percentage of shear fracture, so 100%. I have no idea why this graph includes an upper point of 120%, but never mind. So 100% shear fracture means that material breaks entirely in a ductile fashion. 0% means it breaks in an entirely brittle fashion. It's only relatively recently, this is a fantastic article, you can find it online, that tells you the story. It's only relatively recent that they pull out a chunk of the steel from the Titanic and did the pulling it to bits and look at how it goes ping experiment. And so this is the Titanic steel, and this is the temperature of the Atlantic at that winter. It was particularly cold, even by April that season. And so this is Titanic steel. It had higher percentage of phosphorus and sulfur than modern ship building steel, and you can see why. Because with a ship, what you want is the material to be as ductile as possible. So what happened with the Titanic in that winter was that when it hits the iceberg, basically the material underwent brittle fracture. It did not just go, it went ping. And of course, in those days, ships never carry, as a matter of design, they never carry enough lifeboats to pick up all the passengers. The idea is that ships should sink slowly. They should undergo ductile fracture, and therefore there would always be enough other ships to come and pick up the passengers. The boats are only for transfer, not for taking all the passengers. In this case, the boat just sank before anything else arrived, and that's hence the big loss of life. So this is the ductile to brittle transition in metallurgy. It's relatively well known, it's in undergraduate textbooks. But it's also relevant in rock mechanics, in case there are any geologists in the audience. If you guys are not talking to geologists already, you should, because they really are very good material scientists, except they use totally different words. So this is a schematic diagram I took from this particular paper, and it shows you that if you plot some stress against strain, again, you can just plot force against distance traveled if you like. This is what the curves look like as it goes from ductile to brittle. And the key signature is that when it's brittle, there's a stress overshoot. The other thing that you might notice is that if it is ductile, when you compress it, it barrels. It's a sort of middle-aged body shape. You expand in a little bit. Don't look too closely at me. That's why I wear baggy jumpers. Right, so, but when it goes to brittle, then it either undergoes the diagonal fracture or axial splitting. You actually see a fracture plane. But remember this schematic because I'm going to show you both data like this and also visual data like this. So, what's it going to do in making fudge? Now, if it is not a British audience, I would have given out fudge. But I'm assuming that you all know what fudge is. They use different words for it in America and have no idea why anybody eats anything as disgusting as that in France. So usually when I go to America and France, I bring fudge. Explaining it at customs is difficult. You mean you have a suitcase worth of fudge? Yes. They always think this is just crack or something, but anyway. True is I talk about crack as well. Anyway, so fudge. This is the minimum fudge recipe. Okay, so the reason why I was doing it is that a few years ago I set up this organization called Edinburgh Complex Fluids Partnership. This is our strapline to focus the industrial knowledge exchange with industry of my research group. And we got some money from the EPSRC to work with this Scottish fudge maker. They had some difficulty with how their fudge is setting in the pan. It's great because I get free fudge as part of that work. Right, so here's a minimum recipe. You basically take milk, which for my purposes, I'm going to basically think it is water. It's got some protein in it. You take some granulated sugar, and in fact, these are the only two components I will think about from now on. But there's some butter in there, which certainly at high temperature, it functions as another liquid component. This is irrelevant. I should have taken that out. The vanilla doesn't do anything to the basic physics. So you pour the milk into the pan, bring it to the boil, add the sugar and the butter. You basically keep cooking it. This is the dangerous bit. You've got to, you can boil over and all the rest of it. You keep cooking it until all the sugar dissolves and the butter melts. Bring to the boil, cover with a lid, and then this is the bit that is hard to judge. This recipe that I found says 12 to 14 minutes. The better recipes tell you by a Candy thermometer and measure the temperature, and we'll come to what temperature it is, but it's sort of between 110 and 120 degrees. I'll tell you why that's important in a moment. So remove from the heat, stir the kind of nice smelling bit, leave to cool for 5 minutes. Again, this is an art, how far you let it cool, but the better recipes, again, give you a precise temperature. Then you beat it until it becomes thick and creamy, pour it in, set into squares with a sharp knife, eat. Okay, so that's basically what it is. You basically take a sugar solution and make it incredibly concentrated, cool it, quench it, pour it. So here are two pieces of fudge made in my lab. This thing, most of you know, is called a universal testing machine. I have no idea why no one has taken them to court yet. Because it evidently is not a universal testing machine. You can't test the Higgs boson of particle physics in it, for example. I mean, someone should sue them and make a lot of money, right? Anyway, I'm still waiting for them to sue me for saying that in public, but it hasn't come to me yet. Right, so this is a universal testing machine. So these two have different cooking temperatures, but otherwise cook exactly the same way. So I'm just going to slowly compress it, and you watch. Those are the cooking temperatures. There's a random person walking here in a moment, but he's not relevant. You're a random person walking. I really should put music to this. Is this actually compressing or has it not done it? No, it's now started, sorry. It's supposed to start at the same time. See this? And then this. Remember that diagram? I don't know whether you can see it. I can. There are lots of little fractures here. You may not be able to see it from there. But it certainly does this middle-aged bulge bit, right? So there's no doubt that if you ask a geologist, they will say this is ductile and this is brittle. I mean, what sort of mouthfeel, what sort of words would you think you describe this if I were to give you a piece of this and a piece of that? Shout out. What sort of words would you describe that? It's chewy, yeah? And what kind of words might you describe that? Crumble or crunchy or, you know, that sort of word. I mean, it's amazing how you can look at a movie like that, you can almost feel it in your mouth, right? Go on, say yes. Yes. Thank you, just checking. Yes. Right, so that is brittle, according to that diagram, and that's ductile. And you can look at the low curves, that's increasing time of cooking. And look at those curves, there's no doubt this is brittle and this is ductile. And it's very sharp, 1 degree, and it does it. And it's reproducible. So it's amazing. You know, once you learn how to do it and not burn the darn thing in the pan and all the rest of it, but it's reproducible. So that's basically the summary. If you increase the cooking time, it goes from this kind of what some shops call creamy fudge. There's one particular shop on the Royal Mile in Edinburgh that prides themselves on making the most creamy fudge in Edinburgh. If you increase the cooking time, it becomes this sort of crunchy stuff. And you might see, actually, this is less matte in appearance than this. And there's deep reason for that. That's physics, why crunchy fudge is matte. Okay, so, and you know, they have totally different mouthfeel, which is why it's important, which is why manufacturer cares. If you're aiming to make this, it's no good if this comes out. In either case, they are solids. And so if your recipe really goes wrong, it doesn't set at all and becomes runny liquid. Okay, so that's a sort of fudge taste mouthwash, which is not what you don't want. Okay? So this is what happens on the most basic level. So this is a schematic, but it's more or less correct on some linear scale. But I've drawn out this particular bit. So you need to use a funny scale, but it more or less goes like this, but if I plotted the real data, this bit becomes very crowded and you can't see it. So this is the solubility curve of sucrose in water. This is temperature, this is sucrose concentration. When you start off with, you put a lot of sugar in, and in fact, it's a lot of the sugar sits in the bottom, it's not soluble. When you heat the solution up, like this curve is basically vertical. It crosses the solubility line, and for some time it becomes soluble. Then it becomes dangerous because it comes out again, which is why your fudge making equipment has to be very clean. You don't want any spilled sugar solution that crystallizes on the rim and then the crystal drops in, because then if some little bits of seed crystal drops in, the whole thing crystallizes on you while it's still boiling. But if you're very clean equipment, you get away with it. You follow the boiling point curve. As the system becomes more concentrated, colligative properties and all that stuff, the boiling point goes up. Okay, so it follows this curve until you get to some point C, and then you stop boiling. And this is typically between 110 and 120 degrees. And you quench it. I'm not going to talk about this intermediate stage of waiting. That is about controlling the size of the crystals. But basically, the amount of crystals that comes out at room temperature is this gap. Okay, it's this gap. The distance between C and C prime, the horizontal distance, control the amount of crystals that come out. What you do in the quenching process controls the size of those crystals. That is important, but not all important for my talk, so I'm not going to go into that. Anyone lost yet? If you are, there's no point doing the rest of the talk. No? Not lost yet? Must try harder. Okay, fine. I mean, I must try harder to lose you. Right, so this, in suspension physics terms, that gap controls the volume fraction of crystals in my suspension. For this purpose, I'm going to pretend that the butter is soft enough that it's a fluid, or a very soft solid. So the water and the butter I would consider a fluid phase in this suspension. The rest is essentially sugar crystals. Okay, and that gap is controlled by this T-cook. In other words, you cook it to a higher temperature, you get a more concentrated suspension. Yeah? So that is in fact the only seriously useful thing we managed to explain to the company, that your thermometer is in fact a volume fraction meter. You're controlling the concentration of the suspension by the cooking temperature. So when T-cook goes up, the volume fraction of crystals go up. So that's what it looks like if you put it in an electron microscope. It's not fancy electron microscopy as we heard about this morning. This is easy electron microscopy. This scale bar, in fact, this scale is 200 microns, so the crystals are sort of tens of microns big in a liquid or otherwise soft matrix. So this is a suspension of sugar crystals. So this is the physics of dense or concentrated suspensions. So our hypothesis is that the ductile to brittle transition is in fact controlled by phi, by the volume fraction of the suspension, not by the temperature per se. But of course, if you send that paper to a referee, they would just object because, and rightly so, because changing temperature in this case changes not only the suspension volume fraction, it changes the extent and the kind of Maillard reactions, the Browning reaction that take place. It changes the way the sugar reacts with the fat and with the particularly with the proteins. Okay, so we want a system, perhaps not edible, but a system that is very much the same as this, but we directly change the volume fraction of crystals. That's what our student did this year. We just took a suspension of calcite, is used in paper making, for example, calcite slurries. So calcite, in our case, the crystals are somewhat smaller, but if I don't give you the scale bar, this could have been that sucrose picture. Okay, so here I can actually make, in fact, it takes a very patient students to make these suspensions, because these highly concentrated suspensions are very hard to form into a sample for the universal testing machine. Doing rheology in a small gap rheometer is far easier, but this is her data. So the triumph is actually making these suspensions. It took the order of 3 weeks for her to form a high-density calcite suspension into a shape that looks exactly like the fudge shapes, and all the samples are the same shape. So there are no shape effects here. Okay, so all the shapes are the same. So you just compress it in the universal testing machine. That really is not an error. That's not the axis. The yellow one is not the axis. There really is a small growing up. The next sample down, 0.5, is liquid. You can't put it in the universal testing machine, which proves that it's not a universal testing machine. Okay? So, then I proved my case in public. So it's liquid. You can't do it. And I'll come back to that. So there's a transition between 0.5 and 0.52. That's the liquid to solid transition. The fudge doesn't set, the fudge sets. But in this case, it's calcite. So the calcite doesn't set, the calcite sets. And then it is ductile, is a transitional form, and then it goes brittle. Okay? It looks exactly like the rock thing, it looks exactly like the fudge thing. That's pretty good. I bet you've never seen a graph with rock, calcite, and fudge on it. Same time. It just doesn't happen. Right? Neither have I until today. So, this is just to show you that visually it looks exactly the same. It slowly squashes, it barrels, and then it shows these fracture lines. So visually, and in terms of those graphs, they all look exactly. When did I start?
Just after 3 o'clock, I think.
Right. No, fine. So I do have time. Where is you're sitting there? Because you're going to wave at me, but I need to know where you are so that I see whether you're waving. 6 minutes. No, that's fine, but I need to know where you're waving from me from. Okay. So what's going on? So in metallurgy, people think they know what's going on, although research is still ongoing, of course, but fundamentally, the metals to make the boat the metals are microcrystalline. So in fact, the ductile to brittle transition is about whether these dislocations can move. Now, dislocation is a defect in a crystal lattice. But the trouble is, with my fudge, the sugar crystals are not packed into a crystalline lattice. I mean, the sugar themselves are crystals, but the packing of the crystals themselves is amorphous. It's not in any way crystalline. So there's no corresponding concept to dislocate. I mean, there are no defects. Everything is a defect because it's not a crystal. You get it? I'm not talking about whether the sugars are crystals. I'm talking about the packing of the sugar crystals is not, in any sense, ordered. So it doesn't make any sense to talk dislocation or any defects. Everything is a defect because they're not crystalline. Okay, so, I mean, it seems odd. When we first discovered this, this is very odd because we know about dislocations, but there are no dislocations there. Any guesses of what might be going on there? Just take a stab. Come on, this afternoon, you need to think to keep awake. No? All right. So these are soft amorphous materials, so we don't know what's going on. So it turns out, this is one of those fortuitous things, we were busily doing this, it turns out there's a whole bunch of theorists around the world, unbeknown to me, have been making theories about why there nevertheless may be brittle to ductile transitions in soft amorphous materials from amorphous packing of sugar crystals all the way down to atomic and molecular glasses. And here is one of those theories. It's a bare bones theory. It took me a long time to understand it. When I understood it, I asked them, can I present it in this simple way? They said yes. But I don't know why they didn't present it this simple way in their paper, but never mind. So what they did was they did a simulation, which is a solid, a two-dimensional solid, composed of lots of mesoscopic blocks. These mesoscopic blocks are many, many atoms worth of blocks. And each of these blocks, sometimes in the literature, it's called shear transformation zones, but never mind. Each of these blocks would undergo plastic flow if the local shear stress exceeds a certain value. And that value is called sigma c, some critical value. Now, in their model, the blocks can either all have a very wide distribution of sigma c's, or they can have a very narrow, in fact, distribution, or they can all be the same. Okay, then they apply a macroscopic shear to this block, and that shear gets transferred somewhere or other to be local shear stresses, and each of these blocks can experience a slightly different local shear stress because the block is highly homogeneous and all the rest of it. And what they find is this. What they find is that when they successively more and more anneal their starting configuration, the material becomes more and more brittle. So totally annealed means that, in fact, all the mesoscopic blocks yield at the same stress. All the sigma c's are the same. Oh, okay, and that material is brittle. I mean, it's one of those but of course things afterwards, because if every bit of your material would yield at the same stress, you do something nasty with it, it does nothing, it does nothing, it does nothing. It's slightly elastically deforming, but at some point everybody gives up the ghost at the same time, ping, and that's it. That's brittle. On the other hand, if they make the different parts yield different stress, in other words, the sigma c has a very wide distribution in the material. I mean, it has to be like that. If you get anything else, you've done it wrong. The thing becomes ductile. I'm not trivializing this. It took a long time to arrive at it, but it's fundamentally understandable. So that seems to be the way the field is going. If you're interested in this, the Journal of Physics Condensed Matter, or it's one of the Journal of Physics, has recently got a large review summarizing the discussion at a workshop in Leiden, exploring what's new and what's still not understood in the brittle to ductile transition of soft amorphous materials. And this kind of models are very well developed. Your very own Suzanne Fielding has her own a somewhat different way of thinking about this, but a lot of theorists are thinking about this. So we were thinking along the same lines. So, well, in what way would our material undergo a homogeneous to inhomogeneous transition? So we are thinking that what is driving this is the material is becoming progressively more homogeneous as the volume fraction goes up. Is there any reason to believe that? The answer is yes. So this slide itself can make a half-an-hour talk, so I have to summarize this. This is where the 0 and an infinity got swapped. So it turns out that there has been a revolution in this field in the last 10 years or so. Of course, I only quote my own papers in this revolution, but there's lots of people have participated in it. But it turns out that even in suspension of particles in liquids, at these high volume fractions, the flow physics is dominated by interparticle friction. So we can take a leaf from the physics of dry granular materials. If you take cereal, pour it into a tin, and shake it, the cereal shakes down, yes? That's because when you first pour it in, the friction makes them pack. The moment they arrive, they pack. They stay there because friction makes it stay there. But when you gently shake it, they can pack a little bit better. Okay, so I've got 6 minutes. I think that's enough. Which one do you think is more inhomogeneous? Who thinks to the left? Well, you're paying attention, that's correct. Don't look so worried, it's not a trick question. So that's significantly more inhomogeneous. Some places are very big holes, some places are well packed, depends on how you pour it in. So that is known as random loose packing, that's random close packing. That's highly inhomogeneous. In fact, random close packing is the most homogeneous amorphous packing you can get. And in terms of spheres, that happens at around 54% if they're frictional, and say 64% random close packing. So in suspension rheology, this should be sigma infinity. Sorry about that. So in other words, if you take a suspension packed, the viscosity diverges at this number when you shear them very slowly. The low shear viscosity, that should be sigma 8 to 0, diverges at random close packing, but you shear them very, no, sorry, what am I talking about? No, you shear them very fast, this should be infinity. If you shear suspension very fast, the viscosity diverges at random close, loose packing. But if you shear the suspension very slowly, this should be 8 to 0, the viscosity diverges at a high volume fraction 64. In fact, if you take a 64% suspension and shear it very slowly, if you take a 64% suspension and shear it very fast, it completely jams. That's called shear thickening, because the viscosity has already diverged, the high shear viscosity. And the point is, both of these at 0 shear is a solid, but below that is a liquid. So basically, we can get at these numbers. The point about this, and slightly confusing, the only message you need to get here is I can get at these numbers by how suspension viscosity diverge as a function of shear rate. It's laborious, but it can be done. So we've done it. So in calcite, you've got to trust me because I don't want to give you the entire suspension rheology talk. In calcite, we measure the points of divergence at low shear and at high shear, and measure these volume fractions, and it's about 50% and about 60%. Well, there it is. So below 50%, the thing doesn't jam at all. It's a liquid. And indeed, that's a sample that I cannot test in the universal testing machine. So it is a liquid. Above that is, first of all, is ductile. Now, somewhere along these lines, you expect it to become brittle. You certainly should have become brittle at 60%. Initially, we think it should become brittle at random close packing. But we've never seen that. It happens somewhere in between. And in fact, the reason for that, we think, is any way you make the sample, whether it be thermal or shaping it by hand, always anneals it a bit. It's this kind of shaking stuff. So in fact, the volume fraction that I quote is probably not quite these numbers. When I make the sample, I'm probably annealing it a tiny bit. It probably goes up a bit. Okay, so it's not perhaps wholly surprising, but at least in the calcite case, there's no doubt about it. I'm only changing the volume fraction. I'm not changing anything else. So we think this is the physics. And in fact, we can estimate the volume fraction for the fudge. It's a little bit harder because I need to rely on the sucrose solubility curve. I have to decide how to take into account the butter or whatever. But basically the story is consistent where we think these transitions take place. So let me just summarize. Solid packing of high-density suspensions show ductile to brittle transitions. The underlying physics is almost certainly to do with the samples becoming more and more homogeneous. And this, what we discovered experimentally, purely by accident, because I didn't know this was going on until I went to a workshop, agrees with an emerging theoretical picture of how DBTs happen in soft materials. And I think I have a few minutes left for questions. Thank you.
Thank you very much for a lovely talk. Do we have any? Do we have any questions? Yeah.
Hi, thanks for the presentation. I was curious about the fudge. Would you say it's a colloidal glass where your colloidal particles are attractive to each other? Would that be a possibility?
I think this is the point. I mean, the short answer is yes. The long answer is refer to a very large publication that's now coming out. Okay. But yes, but the only footnote is that fudge or calcite is not colloidal. They don't undergo Brownian motion. They're way too big. It's just the stresses at which all these things happen goes up with particle radius square. So for colloids, it's harder to see. You have to be nastier to the sample to see any of these kind of physics. But yes, in principle. And in fact, if you look at the literature of imaging colloidal glasses using particles big enough for the confocal microscope, embarrassingly including some of my own, we're straying to this physics without knowing it. But I was young in those days.
Hi, I just wanted to ask about sort of the size of your materials compared to the length of the cracks, because obviously if you're getting the brittle fracture, it's because the crack length propagation is able to pass through a certain volume of the material. Is that right?
Yes, although the crack propagation is relatively fast. So once it goes...
But in your more disordered materials, the cracks can't kind of link up across longer distances, right? So they're not able to form that brittle
But it doesn't change the point at which... I don't think it changes the point at which it happens.
No, is that's what's controlling it? That's kind of the question.
What's controlling what?
The length that your cracks can propagate through your material.
No idea. Except that there are people out there in the atomic and molecular world who spend a lot of time stopping cracks. So obviously, if I put an almond in there, it probably stops it. So yes, I mean, I think if I go for grossly heterogeneous things such as large bits of almond nuts in there, it may change it. But then the mouthfeel is, largely controlled by the bits of almond rather than the fudge. So, but yes, people know how to do that, but we haven't started doing it yet. But all our samples are exactly the same shape just to get rid of that dependence.
Would the same picture be true? Sorry, hello. If you had a two-phase amorphous system, so you had amorphous phase dispersed in another amorphous matrix, it doesn't rely on the crystallinity of those sucrose.
Probably, but it's just that the brittle ductile transition of the two phases may be quite different. So, for example, in the background here, I wouldn't mind betting there may be a sugar solution glass in the background too. But because that's a molecular glass, the stress scale of that is probably more than you need to crack my teeth. So it probably doesn't come in.
Yeah.
So,
but it's all about interparticle forces, really.
But it would be interesting to do two amorphous materials or very similar length and therefore stress scales to see what happens there. Then it would depend on the coupling for sure. Yes.
Yeah. Okay. Thank you.
I think in the interest of time, we should probably move on. Or do you want to do one quick one, Andy? Or...
This is maybe a suitable one to end on. So your high temperature fudge, like the one that's more brittle, looks a lot like tablet to me.
No, tablet is different.
Can you make some comment on the physics of tablet as well?
It turns out if you do tablets, that stress overshoot is twin-peaked every time. We don't understand it yet. We have the data. It is very cool, but I have no idea what's going on. But I can tell definitely difference between tablets and fudge.
Well, I think Wilson is around later on, so we could please catch him for any more questions. So let's thank Wilson once again for a brilliant talk. Oh, is that okay? Yeah. And next up, it's my pleasure to introduce Professor Helen Gleeson, who is Cavendish Professor of Physics here at the School of Physics and Astronomy at the University of Leeds. Helen started her career in Manchester, where she did her PhD in liquid crystal optics. And after a brief stint running an industrially focused research unit, she became the first female physics lecturer at the University of Manchester. And then, lucky for us, we poached her in 2015 and joined Leeds. Obviously, Helen is prolific in liquid crystal physics, but also in 2009, she was awarded an OBE for her outreach contributions for women studying physics. And yeah, today she will be telling us about liquid crystal elastomers and their applications. So very much looking forward to your talk, Helen. And I'll waive this 3 minutes before.
So I've obviously got a challenge. Wilson has said that I think I can give at least one really useless fact. Is it not on? I thought it was on. Oh, beg your pardon. Right, my challenge, Wilson gave lots of really interesting facts about everyday things. I'm going to try and give you at least one useless fact that might surprise you during the talk. So, again, I'm going to start in the same way as some of our other speakers and really acknowledge the people who do the work in all of this. I've worked with a fantastic set of students, postdocs, and project students on this, and it's their work I'm talking about. So I'm going to talk about liquid crystal elastomers. I'm going to say, first of all, a little bit about what one is, and then I'm going to describe what a metamaterial is, and in particular, what a mechanical metamaterial is. So it sets the scene for some of the properties of our materials. I'll take you through why liquid crystal elastomers are a new family that have this property. Say a little bit about tuning. I'm going to say a little bit about multi-metamaterials, and then a little bit about applications. So that's how we're going to go through. So everyone in the room will be familiar with liquid crystals. What I've got here is a schematic of a low molar mass liquid crystal system where the molecules are approximately cylindrical and they're all pointing more or less in the same direction. And these are used in electro-optic devices because these liquid crystals are birefringent, so they have optical properties, they interact with polarized light, and they also respond to a voltage. And on the right there, you can see the changes that happen in a liquid crystal device when you apply a voltage. And these have been known since the 70s, and that's not what I'm going to talk about. But the systems that I am going to talk about are liquid crystalline. So this kind of order is important. Okay, so that's what we're going to mimic in the liquid crystal elastomers. So liquid crystal elastomers as a subject has also been around for a long time, not quite as long as small molecules, but nonetheless a long time. And chemically what you do is you take liquid crystal units and you bond them to a polymer backbone to form a liquid crystal polymer. Then you add in crosslinks, and depending on how many crosslinks you put in there, you can make a rubber. And liquid crystal elastomers are really neat. There's lots of very, very nice, fun things that you can do with them. But for the purposes of this talk, again, what I want to show you is that we can make thin films that are bendy, stretchy, and transparent, and have some unusual material properties. Right, so the next definition, metamaterial. So what is a metamaterial? I was kind of highly amused to go to a metamaterials network workshop to find that nearly everything I'd done was a metamaterial in my career, and that's fine. But basically, a metamaterial is a synthetic composite material with a structure that exhibits properties not usually found in natural materials. So there's a definition. It's not my definition, but it's a definition. And these properties, these different physical properties, can be all sorts of things. They can be optical. So that would include things like negative refractive index, holograms, and so on. They could be acoustic, so they could be materials that absorb sound in a very special way. They might be thermal. For example, you can imagine something with a negative thermal coefficient of expansion. So that's a material that when you heat it up, it shrinks rather than expands. And the ones that I'm going to talk about are mechanical. And a mechanical metamaterial has a negative Poisson's ratio. Okay, so let me show you what that is. Take you back to something that everybody here will know, and that's what a normal elastic material does. So if you take a normal elastic material and you stretch them, so here's a block of elastic material, and you stretch it, then it gets longer in one dimension, and it gets smaller in the other two dimensions. Okay, so that is something which has a positive Poisson's ratio, and here's a little bit of maths that tells you how to calculate it should you want to. Okay, but basically, you stretch the material, it gets thinner. An auxetic material, or a material that's has a negative Poisson's ratio, it's a mechanical metamaterial, actually has a negative Poisson's ratio. So what happens then is that as you stretch one of these materials, in one or more dimensions, they'll get thicker. Okay, so that's a mechanical metamaterial. So that's a negative Poisson's ratio. And here are some fun constructs that show you what some common negative Poisson's ratio materials will look like. And these have been known again for about 30 years. So there's lots of really interesting materials that have a negative Poisson's ratio. And I've put some examples up here. You know, from the history, this isn't our work, this is from the history of auxetic materials. So you can see that, for example, they can be engineered from normal materials. You can make a network that has the sort of property that I showed you on the previous slide, where then the bulk properties have this negative Poisson's ratio. And there are a few different examples there. I've shown what the Poisson's ratio is, and you can see it varies from not very much to the biggest one, which is expanded PTFE foam. So, you know, fascinating materials, they get thicker rather than thinner. So what? Well, a mechanical metamaterial, an auxetic material, has some really interesting predictive properties. And I'll come back to this cartoon later in the talk, but you can see that one of them is impact resistance. So if you imagine dropping a ball onto a conventional material, what will happen is that, you know, it'll squish, and all of the stresses are such that it'll get, you know, it's going to get thinner. That's what it does. But if you do that on an auxetic material, those strains are such that the material can push back. Okay, so you get impact resistance. These materials can be shock absorbing, they'll resist fracture, they'll resist delamination. There's a whole range of really interesting potential applications. And in fact, one of the places where you'll find some of these mechanical metamaterials is in Nike running shoes. So some of the Nike running shoes have got one of these structures in the sole. It's quite macroscopic. It's an engineered structure along the kind of shapes that I showed you a few minutes ago, and that helps to absorb impact on running. Okay, so it gives for better running. So there are lots and lots of applications, but I'm not going to go into them. The problem is that these conventional materials that people have known about for 30 years all have this spongy structure. And so that means a number of things. It means that they're quite difficult to make. Some of them are very carefully engineered. And it also means that they can tear rather easily. Okay, if you imagine a bath sponge and you just tear it, it's not as mechanically strong as you might like. So there are limitations in them. And they also can't scale across many length scales, okay, because you can't fabricate them either very small or very large. Of course, nature got there before us, and here's some of my useless facts for you. I think they're actually quite interesting, but there you go. You do, don't you? So where has nature developed these mechanical metamaterials? Well, the Achilles tendon, certain bone structures. This is the little black kitty. She was my cat up until COVID, when she left us age 16. But up until that point, her skin and other cat skin was auxetic, okay? One of the other examples I really quite like, but I haven't put a picture up, is the teats on cows are auxetic. Okay, and probably, I'm not the bio anything, but probably because one of the properties of an auxetic material is its tear resistance. Okay, so there are loads of really interesting examples in nature. Right, so let me go back to our liquid crystal elastomers. So here you go, there's our liquid crystal polymer. We've shoved a few crosslinks in there, and so we've made our liquid crystal elastomer material. And that's what we make it of. And actually, the way we make it is we put a lot of these reactive mesogens, these small molecules, into a liquid crystal mold. We use normal liquid crystal methodology to make them all line in the direction we'd like them to, so we can make a very uniform thin film that's typically about 100 microns thick, and then we can play around and stretch it. And it turns out that these liquid crystal elastomers have a glass transition below room temperature, which is nice because they're nice and stretchy at room temperature. And they are optically and mechanically isotropic. And there was a very different reason we wanted to make these. And I'm not going to go into that because that really is a different story. But it was Divesh who came up with this formulation and started testing the materials. Okay, so I've said all of those things. That's basically my view of what our materials look like. So one of the experiments that Divesh did was that he measured how thick the films were when we stretched them as a function of strain. So what you can see, you'll see a number of these graphs, and I just want to sort of point out the important thing. So this is as we pull them in a direction perpendicular to where the liquid crystals are lining up, and this is what happens to the thickness. And you can see they do start by getting thinner, and then they all start getting thicker again. So there you go, it gets thinner, thicker, thinner, thicker. And there are different materials, the different materials have different properties that, different amounts by which they get thicker, different strain thresholds at which they start to have this negative Poisson's ratio. So what's going on? Is our liquid crystal just like the PTFE foam? Is it porous? Well, we did lots of experiments, and the answer is no. Okay, not even when we stretch it. So this is happening at a molecular level. Again, another really talented researcher that I was working with, Tom Raystrick, did work on measuring the order parameter, and we found that as we stretched these materials, the liquid crystal order, the uniaxial order, reduced as we stretched it, but the biaxial order increased. So that gave us a big clue to what was happening. And in fact, if you think about what's going on, if you start from a system where the red shows you where the liquid crystal's pointing, we're pulling it in this direction. In these systems, the molecules pop out of plane. There's a biaxial deformation, they pop out of plane, and that's why they start to get thicker again above a particular threshold. So we were it took us a while to work out. We were very pleased to see it working. But of course, a lot of this was on thin films, and can we actually see it? So this is on 100 micron thick films. And so we made a material where the molecules are pointing in this direction, so they're pointing towards you. And if we stretch it, what you should see is that, first of all, this thin film, this is of the order of millimeters and centimeters. First of all, it'll start to get thinner, and then it'll start to get thicker again. So let's hope the movie works. There it is getting thinner. And there you go, it's getting thicker again. So you can see it's really nice and stretchy. It stays transparent. It's the only transparent auxetic material, synthetic auxetic material, which makes it interesting for other applications. And we can prove that it's biaxial by doing a kind of microscopy called conoscopy. So it's not, the order parameter measurements are always tough and not completely direct. We make lots of assumptions, but we could show that these really are biaxial systems, and that's exactly what's going on. So we were very happy with that, knew what was going on. We could make the materials, we could get high negative Poisson ratios. We knew what direction the auxetic response was going to happen in. We knew what the mechanism was. We knew that it happened at a molecular level, which was all great, and our system stayed transparent. So happy days. And we patented it, and I'll come back to that just before the end. What can you do to tune it, to get different properties? And I'm going to go through these slides relatively fast. A lot of very good work has been done by, again, members of the team, showing that, for example, if you change the crosslinker density, you get differences in the magnitude of the Poisson's ratio, but also the point at which you start getting the auxetic response. This is really nice work where Stuart Burrows has changed the spacer length. This is a new X-ray kit in Bragg. So we can see that as we change the spacer length, we actually go from pneumatic to a different kind of liquid crystal phase. And again, you can play around with what's going on with the auxetic response if you do that. The threshold depends on how far away you are from the glass transition temperature, it turns out. You can do, I can't do, people can do clever chemistry to actually make multiple layers of these materials. So this is a nice pet raft work that means that you're not stuck to just 100 microns, but you can start to make thicker amounts if you want. And you can start to tune the optical properties. So if you want to put these materials between glass so that your glass is stronger or fracture resistant or whatever, then you have a chance of doing that. And this, again, just shows how we can tune the refractive index by changing the composition of the material. I mentioned delamination resistance, and you can see if you actually make one of a sample where we've dispersed silicon materials in there, you can see, and then strain it, you can prove that delamination resistance is real. So there's lots of, all the stuff that was predicted turns out to be exactly as we were hoping. But this was the one that it took us quite a long time to show. So remember the cartoon where you bounce a ball onto a conventional material, steel ball, drop it on, and there's nothing that stops the impact, there's nothing that pushes back, and then there's our auxetic material. So we finally got round to reproducing this, and I'm going to show you a video where we drop a ball on each of them. Okay, and there's a bit of glass under each of these. So there's polyurethane, there's glass, polyurethane film, and in this, there's a bit of auxetic film in between the glass and the polyurethane. Oh, hang on. I'm hoping that movie shows. Thank you. There you go. So, yeah, the glass smashes when you don't have the auxetic material. So this is exactly what's going on in that system. Now, I know I'm going to finish in just a moment. I want to do a couple of shout-outs. So I mentioned multiple metamaterials. And Tom Roestrick, who is currently a postdoc with me, about to take up a lectureship, and one of the postgrad students I was supervising, Tom Morehouse, decided that they should try and make a diffraction grating in the auxetic material and then see what happens. And there you go, you can certainly do it. And when you strain it, if you then do AFM on it, so this is showing the pitch of the diffraction grating here, this is unstrained, the pitch is about 50 nanometers. Strain it, the pitch gets a bit smaller, that's what you'd expect. But actually, if you strain it further, the pitch actually gets bigger. And that's because it's auxetic. And this is on a 10 nanometer scale. So this really is happening at a molecular level. So really neat thing to show. So I mentioned we patented it. We patented it in 2017. It's quite hard work doing commercialization. But we've done quite a lot of work on it. We've gone from small samples to really quite big samples, won funding, spun out the company. We're at the next stage. So if anyone out there has got £1 million or something, they don't know what to do with it, please come see me afterwards. I can suggest some things. But what we're hoping to do, is to use this kind of film in numerous applications. And one of them is to give enhanced protection to wind turbine blades, where there's a big problem with their lifetime because of erosion, because of the raindrops hitting the leading edge of the wind turbines. And I'll leave that for an exercise for homework. If you just work out the velocity of the turbine tip going round, you can begin to see how fast anything is actually hitting it. And it really affects the lifetime of those blades. So that's one of the applications we're hoping that we'll be able to make a difference with. Obviously, we need to make big sheets if we're going to do that. Right, so let me draw to a close. And I guess it's just saying a few things about stuff, right? The fundamental stuff that we did, the fundamental work that we've done, was what allowed us to understand the mechanism. So lots of, fundamental liquid crystal physics, polymer physics, and so on. Right at the beginning, I said we never set out to do this. It was a synergistic discovery. We were actually thinking of doing something else, and it was a really off the scale sort of. Do you think you can make mechanically switchable focus lenses for cataracts? That's what we were trying to do. Still haven't tried that bit. You get lots of new ideas from doing things like that. There's lots of things still on the to-do list. I think the team's done wonderful work. We would like to look and see whether these are also acoustic metamaterials. Liquid crystal elastomers are inherently sticky. And so can you combine the adhesion with the auxetic properties? That's something we're quite interested in. Obviously looking at other formulations. And I just want to finish with a shout out to the team and the facilities at Bragg. We have great people here at Leeds. We have great facilities, great collaborators, and I don't think I would have ever done this work at Manchester. I know you mentioned I moved from Manchester, and Leeds is a special place for that, and so thank you to everyone at Leeds for allowing us to do work like this. There we go, thank you. Oh, and thank you to the people who give us money. Yes. Thank you.
Thank you so much, Helen. Lovely, lovely talk. Jill, we have time for a couple of quick questions. Edmund.
So could you say a bit more about the challenges and how you might approach the scale-up? Because that obviously is the big thing.
Yeah, so the biggest challenge is that actually our current formulation, what's important is you have to have a liquid crystal phase before you polymerize it. And that's quite hard to engineer. Right, that's a materials formulation thing, and that's not where my expertise is, but it turns out to be quite hard. So what we did was we tricked the system by putting low molar mass material in there that we then wash out when we've made the polymer. And that's fine because it works on small scale, but that washout means that your system shrinks. And so if you want to then scale up to reel-to-reel processes and things like that, it's really difficult to do that because you've got waste and you've got the added problem of the shrinkage, which is really, really problematic. So we've submitted a paper where we don't have to include washout material, so that's going to really help. But the big scale up is, can we make it on a reel-to-reel type process? Because we make it. My background is liquid crystal devices, and that's how we started making it. Well, that's not how you're going to make these in big sheets.
Any burning questions? No. I think in the interest of time, we'll move on. Thank you so much, Helen, again. I'm sure Helen's around if you want to chat to her later. Sorry. And last but not least, we've got Dr. Rosa Catania from the School of Chemistry, where she's a research fellow in membrane protein biotechnology. Rosa has had a kind of international career. She's moved around from Italy to the University of Missouri and then did her PhD in Nottingham, where she stayed on. She was awarded an EPSRC Doctoral Prize Fellowship and then moved to Leeds in 2020. Her work now focuses on kind of bioelectrocatalysis and kind of protein reconstitution in kind of hybrid lipid polymer nanostructures. So I think she'll be telling us more about that now. So over to you, Rosa.
Thank you, Arwin, and thank you to the Bragg Exchange to give me the opportunity to present some of the recent work that I've been doing in Paul Bill's group in the School of Chemistry. And I'd like to start by introducing what hybrid vesicles are. Hybrid vesicles are something in between liposome vesicle made of pure lipids and polymerosome vesicle made of pure polymer. So if we have a closer look to the membrane of this vesicle, they are a mixture of phospholipids and amphiphilic blockopolymers. There are a variety of blockopolymers that can be used. In the group where I work, we've been using this specific polybutadiene PEG blockopolymer. we have fully characterized. We have looked at the homogeneity of the membrane and also of the thickness of the membrane, of the bilayer of this vesicle. Something that these hybrid vesicles are really good for is membrane protein reconstitution. So if we use this vesicle to reconstitute membrane protein, we gain the biocompatibility from the lipid side and the stability from the polymer side. So we have shown in the past that, for instance, if we use a molar ratio 50-50 polymer to phospholipids, we can retain a high level of a membrane enzyme reconstituting into the vesicle while we gain a long-term stability. And when I say stability, we are talking about many months, even years. So it's something quite impressive for this type of vesicle. And for those that may not be familiar, usually when we do reconstitution of membrane protein, we use detergent methods, which means there are lots of detergents involved, steps, it's quite a tedious procedure. Where actually we have designed new detergent-free reconstitution methods for this hybrid system, which made use of another amphiphilic blocopolymer, styrene maleic acid blocopolymer. And here I have a snapshot of how the system works. We isolated the membrane protein with the SMA copolymer. We mix with the hybrid vesicle and expose it to magnesium chloride, which allow the SMA copolymer to precipitate, and so we have reconstitution into the hybrid vesicle. We have used our cytochrome B03 enzyme. This is a terminal oxidase of E. coli inner membrane. What this enzyme does, it reduces oxygen to water while oxidize this quinolone molecule here. So we can look at the oxygen concentration in solution to work out the activity of the enzyme. We compare the detergent method here in gray, where we have isolated the enzyme into detergent micelles, so we have quite a high activity, and then we reconstitute it either in hybrid or liposome as expected. But when we use the SMA method, our new developed method, we can see we still have a quite high activity from the SMA extract, which retain the activity level when it's reconstituted into the hybrid vesicle. Why surprisingly, we couldn't find any activity into the liposome. And this is why actually we didn't have any reconstitution into the liposome. So we look at the gel of the reconstituted sample, why in the hybrid vesicle we can see the band from the membrane enzyme with a quite high reconstitution efficiency. So this method is quite specific for hybrid vesicle. We also want to make sure that we have transmembrane reconstitution. So we use a pH-sensitive dye that will change fluorescence based on the pH of the vesicle. And we can see that the proton, we exploited the proton pumping activity of the enzyme, where we can see an increase in the inner pH, which means the enzyme is in the same orientation as we would expect in the natural membrane of bacteria. Very briefly, just to say, we have applied this method also to crude mixture of membrane protein. So if we have all membrane protein from E. coli, these are the one extracted with MALP. And when we expose to magnesium chloride and precipitate, there are no membrane protein left in the supernatant. But if we have the hybrid vesicle, we can see that membrane protein are retained in the supernatant, because they have been reconstituted into the hybrid vesicle. So now that we have this direct and quick method to reconstitute membrane enzyme, of course, there are a variety of applications that the system can be used for. Here I give you an example, one application we are currently working on, which takes inspiration from nature. Nature used this membrane redox enzyme to catalyze important reactions that can be used for energy conversion. So we can think of using these membrane enzyme reconstituted here to build up biofuel cells, for instance. So how do we go from here to the biofuel cells? Well, we have done lots of work. I would just briefly say that we have optimized gold electrode interface here for the hybrid membrane to form a flat hybrid membrane here. We've used bioelectrochemistry to check that we have diffusivity of this quinone molecule into the hybrid membrane. Some impedance spectroscopy to check that we have full coverage of this gold electrode. Now, if we have the membrane enzyme reconstituted into the vesicle, we can see that the oxidation peak of the quinone molecule disappear, and we have this peculiar shape of the cyclovoltammetry, which tells their currents is driven by the redox enzyme here. And also we maintain good impedance spectroscopy. We maintain good coverage of the electrode. So we can actually build a cathode side for our biofuel cells with this system. And we also had a sample longer than, like even older than 1 year, and they still show activity and ability to cover, the enzyme. So there is lots of potential in terms of storage for bioenergy application. But we want to look a bit more about using other polybutadiene PEG polymer. So we selected a variety of different polymer where we change the length of the polybutadiene part, so the hydrophobic parts here, and also we change the PEG here. We selected those that we would expect to give us bilayer vesicle, and we confirmed this via cryo-EM. And we follow this procedure here where we use a few fluorophore to sort of control the total amount of membrane protein we would have in the reconstitution, and another fluorophore to tag the E-stack of the enzyme so we could gain information about the orientation of the enzyme. And I'm about to show quite a large set of data. I will not go into the detail, but just want to give you an idea of the sort of different trends that we can see. By changing the combination of this polymer mixed with POPC to form hybrid vesicle, so we gain a range of different vesicle size, but still in the liposome range. And we can see that there is quite a high impact into the reconstitution efficiency of the system with a copolymer with a shorter hydrophobic chain give us a higher reconstitution efficiency compared to the longer one. We can see a difference in the orientation of the enzyme when it's reconstituted. So we have, for instance, these polymers here that give us a more homogeneous orientation into the reconstitution. This has also an impact into the activity. So if we compare the electron transport of this cytochrome BO3, we can see that some enzyme will perform better in this area of the polymer we've used. And we have done more control, so more other work. And what we're doing now is we're trying to gain some knowledge and draw some more conclusion in terms of which parameter affect which properties, so we can design bespoke system. For instance, if we are more interested in a higher reconstitution efficiency, or we are more interested in stability and activity, then we can select the right polymer to use for this system. So we show you our new methods for the detergent-free reconstitution. I show you one application we are working on for bioenergy application, and the screening we are doing to gain more insight into the different polymer for hybrid vesicle. And I would like to conclude just to say thank you to Paul, all my other supervisors, the groups where I work on, and of course the funding, and thank you all for listening.
Thank you very much, Rosa. Do we have any questions? Was that a hand? No. One there?
Thank you. Can you go back to the first page? I mean, the first slide. Can you go back to the first slide?
I think so, yeah. Sorry about that. It's going to be quite a journey. Back?
Maybe next. And then, yeah, I'm just curious about the lower left image. Why it's not clear? I mean, why the image is not so clear?
Why is it not clear? Oh, sorry, I just cropped it from one of the recent publications from the BILS group. So if you check the actual paper is in high resolution. I don't know if it might be a bad resolution for me.
I thought it was your results. I thought it was your results. I thought it was your data. So.
Sorry, I missed that.
Yeah, I thought it was your data.
Oh, my data. Oh, no, this is actually from one of Paul's PhD students that has done the cryo-EM. So maybe if you want to check the...
Okay, thank you. So, understand.
Final question for Rosa. I've got a question, but I'll ask you over wine because I think most people might be a bit thirsty by now. So with that, please let's thank Rosa and all our speakers for this session, for a fantastic soft matter session.
Okay, that pretty much closes the talks for the first day of the Bragg Exchange. I've got a request from the team that if you snuck in late and you haven't picked up your name badge, please can you make sure that you get it so that we can make sure you are registered. That helps us to track our evaluation and impact. And also if you need any form of a letter of attendance, then we can only do that if you've actually registered. So please make sure you get your name badges. And you'll also remember that your poster voting slips are in your name badges, so please make sure to use them over the next 2 sessions. We're now going to drinks, posters, and exhibition. Please do join us for that. There's another hour and a bit to check out the posters, and there is a selection of drinks and nibbles. So, thank you.
you.